arrangements this morning, and the arrangement is such that we had already scheduled the second lecture to be the molecular biology. Ademola, this shower is from called University Teaching Hospital. It's an honor to have you there. Welcome. It promises to be an exceptional session. Thank you. I can see MLS Akinlabi there. And Sir Job, I believe the audio is fixed now. You should be able to hear us. If you can hear us, you can just react. You can use the reaction tools. The audio is out here, and we can see it. All right. Ooh. So yeah, I said many of us should tell us what you intend to gain from the session, maybe one sentence of what you look forward to gain, but I've not yet seen that. It's good to see you, FMC Abeokuta. That's Israel Ziani. Thank you so much for joining in. It's an honor to have you here. I could see Adeyemi Adetola. It's an honor to also be here for joining. Promises an extraordinary session. And our panelists, I believe they're going to be joining the call shortly. Okay. Yeah, Adif Kemi, thank you for that. I know enlightenment is going to be the first target of this session because many of us as medical laboratory scientists, we practice. I can remember during the undergraduate level where we've been trained in medical legal parts of this session. I think it's just one, 2 or 3 unit course. That's the only time we did something medical legal. The more medical ethics, I think it's medical laboratory ethics and legal aspects. I tried researching that period, though it's a bit further a year back, to see documents that really back ethical practices or legal practices in the profession or legal implications of some of our practices in the profession. I can't remember the questions the lecturer gave to us that time, and he told us to go and research on the net. On going for that research, by the time I came back, I just discovered that I couldn't get enough resources that really, target the laboratory practices. So these are part of the enlightenment I know we're going to be having, because those who are coming to take this session to us, or they are releasing this to us, are professionals that are practicing. And beautifully, this person is also okay. So this legal practice we are coming to experience is uh, is going to be an enlightenment for myself, because this will be a first-hand detail from a professional. Now, another thing I want us to also look at is, many of you will agree with me that what we add in training and in practices is fewer exposure to the molecular practices or molecular diagnostics of medical laboratory practices. But now we have a professional who has qualification in molecular biology and genomics, and is ready to even share insight to us. I could remember while we were also in school and we were dealing with the bioinformatics, I see some of us, when we went into it, it looks so gibberish that we don't even understand what it is. I could remember all those DNA sequence. I could remember when we tried to get the phenotypic models of the organisms we are trying to, synchronize, or you are trying to confirm. I could remember the pairwise alignments that many don't even know the significance of those things that year. You go into the database, you don't know what it means. And this is what this seminar or webinar is trying to target. It's trying to bring us to be up to date with the global practices in the profession, as well as the legal session. And I'm glad to tell you that our our lecturer is already set, and they will soon be joining us in the next few minutes. About 3 minutes time, we're going to be seeing him come live. I can see that. So I see MLS Paul on the call, but I don't know, maybe he's set to take a session. So we have the way it's being scheduled, as according to what is laid in my hands today. We have the first speaker. That's MLS Kemiki Olalekan Ademola, and is the laboratory manager, Babcock Central Research Lab. I also have with me on this same call right now, as a member of the panel, and that's MLS Afolabi Rashidat Ibiroke. So also on this same call, I have MLS Yusuf Ahmed. She's also on the call and is part of the team that are making this possible to us. So I give a special shout out to the biomedical team, week team, for this our own very own annual general meeting. And she's also the secretary of the committee for this program, and that's MLS Yusuf Ahmed. It's an honor to have you here. I could see Horizon Health, but I don't know who bears that name though, but okay. All right, so that being said, our second speaker, who is also going to be coming by 12, is also the CEO, Zenith Diagnostics Limited, and MLS Paul Oreji. Sorry if I don't pronounce that name right. Just call me to order. We also have Daniel Oladile on the call. So we also have Daniel Oladile on the call. I can see Daniel Oladile. It's good to have you there. Okay. MLS Modupe, can you hear me now? At least for the meantime, till we rectify the digital audio. Okay, so I think it's better now. Are we good? All right, thank you, everybody. So now, we are trying to marry our goals, and I've been seeing many medico-legal, because many wants to know the legal implication of their practices. Somebody said to gain more experience. I wish I could talk extensively on experience, but that's also a beautiful one, to see experience. So we're going to experience newer things today. Okay. I want us to do a kind of, I will call it a pre-test assessment. And what I just need you to do is you reply with a number. So before this session, so we are starting with the first question that I'm asking is this: how will you rate your knowledge of the bioinformatics and molecular tools in medical laboratory practices? How will you rate it between 1 to 10? So just drop your own rates. I'm going to be pinning the posts. So that everybody could see it. So how will you rate it? So let's have your rates. Okay, Selimon says 5. Okay, Olariwaju says 2. MLS Job says 3. This is a sincere, I love the sincerity that is being revealed by everybody. That's a good one. 3. Oh. 6. I love that. Michael Makinde, amazing. We have somebody that's already above average. Oh, Oladebo, 2. Precious. I see. 2. Wow. Daniel Oladele is saying 2. It's online. Ojeda Pomesi, that's 2. 5. 2. Ooh. Thank you, everyone, for this sincerity. So, in average, from what I've been looking at, majority of us at this beginning of the session doesn't really have the exposure on the molecular diagnostics. So we are moving ahead, and the person that is coming on board is our facilitator, first speaker, MLS Kemiki Olalekan Ademola. But before then, you will hear his citation that will be ably read by MLS Afolabi Rashidat Ronke. Over to you, ma'am. Thank you. I don't know, maybe MLS Afolabi could hear me, but we cannot hear her read the citation. So please, in the bit we are going to be starting, it might be a technical glitch from our end, and it might be some slight challenges. So please, because our lecturer is all set.
Do you want me to start again, sir?
Is he on the call now? Oh, please start again. Thank you so much. We just could
Okay,
hear
all
your
right,
voice.
sir.
We couldn't hear your voice earlier. Thank you so much, ma'am.
All right, sir. All right.
We are not hearing you again.
Hello, sir. Can you hear me?
We can hear you now.
Okay. All right. Let me go again. Mr. Kemiki Olalekan Ademola.
I guess it's the network. Sorry, I
Hello,
believe
sir.
there
I can
must
hear
be.
you.
I can hear you now.
Can you hear me now?
Yes.
I'm taking the citation already. Okay, let me try and go again, please. Mr. Kemiki Olalekan Ademola is a medical lab scientist, molecular diagnostic scientist, research faculty member, and laboratory manager within the Research, Innovation and International Corporation. He is a directorate of Babcock University. He currently manages the Babcock University Central Research Laboratory, where he provides scientific, strategic, and technical leadership in multidisciplinary research support, laboratory operations, quality systems, equipment coordination, professional training, and institutional research capacity development. He is a certified African genomic medicine trainer and the chief executive officer of Purging Molecular and Bioinformatics Solutions, a platform advancing molecular diagnostics, genomics, bioinformatics, laboratory development, assay design, research support,
A switching.
equipment and reagent sourcing, and locally relevant diagnostic innovation for Africa. He has trained hundreds of healthcare professionals, scientists, researchers, and students in PCR, qPCR, molecular diagnostics,
All right. Mickey, I believe the network glitches didn't allow our citation expertise or professional to be able to read it to the end. However, I will pick it up from here. just to conclude that Mr. Ademola is a published researcher whose scholarly contributions span antimicrobial resistance, infectious disease genomics, molecular cancer multiomics, machine learning, molecular docking, and precision medicine. His professional development includes programs delivered through McGill University, Welcome Connecting Science, H3A BioNet, and the African Genomic Medicine Training Platform. He is affiliated with several professional and scientific organizations, including the Medical Laboratory Science Council of Nigeria, Association of Medical Laboratory Scientists of Nigeria, African Society for Laboratory Medicine, African Society of Human Genetics, Association of Antimicrobial Resistance of Nigeria, and Bacteria Vaccines Network. He is passionate about repositioning medical laboratory scientists as molecularly competent, AI literate, genomics ready, and innovation-driven professionals capable of developing sustainable diagnostic solutions for Africa. Please allow me and permit me to introduce to you MLS Kemiki Olalekan. Thank you. You can go ahead, MLS Kemiki. We can't hear you, sir, in case you are speaking. I guess your mic is muted. Try to unmute, sir.
Yeah, good morning, everyone. Okay. Thank you so much. Uh, you see, I paused a little bit because I'm trying to be official with my brother. Thank you so much, MLS, and thank you, everyone. Sorry, I think I had a little glitch trying to share my screen, but I think that will be sorted now. So I think let me begin. Let me try to share the screen. So sorry, just give me a few minutes and then we'll have the screen ready. So okay. Yeah. Please, can you see my screen now? Please, can you see my screen? Someone please respond. Can you see my screen? Anyone see my face? Network issues. Please, can anyone see my screen, please? Can you unmute and let me know if you can see my screen? Please, can you see my screen? Mr. Temiloluwa, can you see my screen, please? Okay, not yet. Okay, so I think I can. Please, can you see my screen now? It's going to be a long day. Okay. MLS Temiloluwa, please, sir, I've uploaded my slide already. Can you just help me share it so that I can just go on? I think I'm having issues sharing the screen here. It's not working. It's not working, but the slide is online, so we can all see it together. Can you kindly help me with that, sir? It's showing that my screen is already showing at this side. Okay, let's see if I can do this. Can you see my screen now? Can you see the screen now, please? Can anyone see the screen now, please? I believe the screen should be. Yeah, he said that you can see my screen right now. Can anybody just see it? And bioinformatics emerging tools for improved healthcare delivery. Can anybody see it now? It's going to be a long day. Okay. This is a lot. I don't know what's happening. Hello, brother me. Can you help me share my screen? I have. Nobody is saying they can see it, so I'm afraid. Oh, you know, the screen is that once I'm looking at that screen, I can't see what you are saying at the other side. Okay, so no problem. That means that I'm audible right now, right? Okay, thank you. Sorry, I was already frustrated a little bit. Thank you. Yeah, thank you, boss. Okay, so good morning, everyone. Good morning, everyone. It's a great privilege to be here this morning. I would have loved that at least I can hear back responses. Maybe some one or two persons can unmute so that I can know that I'm not alone in this room. If you can do that, I would greatly appreciate that because I can no longer see the streaming platform. But I believe we are going to have a great day together. So I am Olalekan Ademola Kemiki, and I will say it's a great privilege for me to be here today. So I want to start by appreciating the Oyo State, sorry, for inviting me for this program.
We thank
Uh,
you.
Thank you, sir. So I believe that we are going to have a great time today. So I'm going to be talking on molecular diagnostics and bioinformatics, emerging tools for improved healthcare delivery. Before I go straight into the lecture, let me break it down. We want to be looking at molecular diagnostics and bioinformatics in the way in which it is improving the current healthcare delivery in the world. So let's dive right into it. So at the end of this lecture, you should be able to understand why molecular diagnostics matter. You must be able to understand molecular targets. You must be able to choose the right method for analysis. And then also you must be able to transcribe results. You must be able to transcribe data into clinical results that can lead to clinical actions. So let's go straight into it. I'll take my time to explain molecular diagnostics. I'll go through a lot of forms of molecular diagnostics. Then I'll talk about diagnostic bioinformatics. And then after that, I'll talk about genomic surveillance, and then I will end with quality control and ethics. So let's go straight into it. The diagnostic paradigm shift. When diagnostics started, let me use the word diagnosis. When diagnosis started, uh, doctor needed to listen to patient's narrative. That's when you go to the hospital, the doctor will say, okay, sit down, explain your problem to me. So there were days when diagnosis was all about listening to the patient and bringing out the solution to the patient's problem from the way the patient is talking about his problem. That used to be what is called patient narrative. It still exists today, but it's not enough to make an inference from patient narratives alone. It will be wrong, because even the patient cannot adequately put in words what they are feeling medically. So to compensate for that, there was the advent of traditional diagnostics. Traditional diagnostics is when we started with a phenotypic method of diagnosing. It was phenotypic method of diagnosing. They check you, look at okay, how you feeling, check the temperature, is there color change, check the eye. They said they check the eyeball, they check the eye, is it white, is it yellow? All those ones, they are phenotypic mode of diagnosis. Then after that, we move into what we call the culture-based diagnosis. That was when the advent of microbiology came in, that microorganisms could now be cultured, and then after cultured, we could identify them and probably profile a solution to the problem. I'm sure our medical microbiologists, they are doing very well in this field. Then also serology came in. Serology came in. The advent of measuring individual elements also came in. Enzyme-linked immunosorbent assay, who happen to be the greatest in that era, also came in, where we get to identify antibodies produced by pathogens. And then we measure the antibody in any way we like it, direct, indirect, whatsoever we like. But what is the characteristics of these the traditional diagnostics? It's very slow. Imagine if you have to culture tuberculosis that takes about 6 weeks for you to culture, the patient would have died. Even currently, one of the issues that is happening in clinical practice is the turnaround time with our results. Imagine that you have to culture to identify a resistant bacteria. Before you do all of those culturing within 3 days and things like that, a lot is already gone in the life of the patient. Now, also, it requires that you have culturable pathogens. You have to have pathogens. If you don't have pathogens, if the pathogens are not culturable, so many organisms are not culturable. So if you buy a media, you are just busy wasting your time. And then also, one of the challenges of that era is that era couldn't predict what is likely to happen to an individual. You can't predict. Even though you are running what's it called, clinical chemistry test, you are only seeing what's already going on in the patient. You cannot predict what is likely to happen to the patient before it becomes that level of obtainable or feasible diagnosis. Now, also, you could see here that I said the struggle with early cellular stage, cellular changes. You can't do that. But there's now a diagnostic paradigm shift. Traditional diagnostics have now been complemented, not replaced, please. The essence of molecular diagnostics is not to replace traditional diagnostics. No, it is there to complement traditional diagnostics. Molecular diagnostics has come in now. This is when we begin to analyze samples based on the DNA, RNA, or protein presence. So there's a shift now. We could say this person is carrying Mycobacterium tuberculosis, but right now we could go far and say it's not that he's only carrying Mycobacterium tuberculosis. He's carrying an antibiotic-resistant Mycobacterium tuberculosis, maybe rifampicin, RPOB. We can go that far because we now have molecular diagnostics. You are thinking that, okay, somebody is suffering from elevated amount of ACE. You know, ACE, which is a very, very important enzyme in the kidney, in the work of the kidney. And the person is saying, okay, this person is having the EUCR is elevated. We could see that the electrolyte is off grid. A lot is happening. But what is the underlying cause for all this abnormality? You can trace it down and say, okay, it is the ACE gene. The molecular diagnostics can answer that question, that this abnormality is sponsored by ACE gene. Not only is that it's sponsored by ACE gene, we have found out that it's a mutation in ACE gene that is sponsoring this. So molecular diagnostics has helped us to answer more questions. Molecular diagnostics have really, really helped us to answer more questions because the questions have now changed. The questions that we are answering as medlab scientists have changed. We are no longer answering the questions that we were answering before. I believe in my next slide, you begin to get all of these things together. What is the characteristics of the molecular diagnostics? It is rapid, it's very fast, it's highly specific and sensitive. You don't need to culture. That is why we have what we call metagenomic sequencing. You don't need to culture anything before you know what organisms are present. You don't need to culture before you know if the organisms are viable or they are not viable. And then also, one of the things that this can do for you is that you are able to identify minute genetic mutation even before any clinical symptoms appear at all. So there's a lot that can happen. There's a lot that can happen before we begin to see any change whatsoever. So let's go. Let us talk about the landscape of diagnosis. I'm sure that I've been in practice for about 10 years now or more. I think 10 years or more. So one of the things that when I came into practice, our role was just to diagnose. How do I mean? When a patient is undergoing a particular clinical symptoms, they send a sample of that patient to the laboratory. And then what we do is to find why that patient is acting the way the patient is acting. That was the landscape of diagnosis that period. But right now, the landscape of diagnosis have truly changed. Now, people come to the lab to predict if they are going to have a certain disease. Before, it was when the disease have come, they come to us. Right now, they come to us even when the disease have not even shown face at all. So the laboratory is acting as an hub where you can predict the presence of a disease or the likelihood of developing a condition. Let me give us a very good example that we should all know about. It's the story of Angelina Jolie, the actress. She found out her mom died of breast cancer, and Angelina was a little bit worried that she's hoping that she will not also die of the same breast cancer. Do you know what Angelina did? Angelina approached the lab, and when she approached the laboratory, they ran a test for her. And when they ran a test for her, they were able to discover that Angelina has that gene, but the gene is not yet expressing. That is, she has the potential to also have breast cancer, but what will ignite the expression of the breast cancer have not been activated. Do you know what Angelina did immediately? She removed her 2 breasts. She wasn't interested. So the lab is answering questions that we were not answering before. Now they are asking us, I want to know my status. Am I a carrier, or do I actually have this disease? Also, this landscape has changed. Now we are screening. When I say screening, we are busy finding disease that are not even shown up at all. The first one is that we are predicting. Now we are screening before there's a major clinical deterioration. That is what we are doing with HPV DNA. When you are doing HPV DNA, you go, you screen, you check, are you sure that you don't even have any of those serotypes of HPV? So after you've done the screening, then you now return back to get the vaccine. So you see that the landscape of diagnosis have really changed. When a child is given birth to now, normally screening should take place. When the child has been given for, you check out for the alpha fetoprotein. That's what they do in chemistry, because that's a protein expression of something. But we will just go dry into the gene and see, is there any change in the gene? Is this person predisposed Does this person is screened for trisomy syndrome and things like that? So the landscape has changed, but we are still diagnosing. But now we are diagnosing and classifying. We are not just only detecting diseases again, we are detecting the disease and we are now classifying them. You say, oh, this person has TB. This is the type of TB that this person has. Okay, this person has HIV. It is either it's HIV-1 or it's HIV-2. This person has HBV. This person has leukemia. And we are now saying, okay, this is the leukemia that this person is AML, is CML. There's a lot that the lab is doing currently. Then also the landscape has changed. We are now guiding treatment. How do I mean when I say guiding treatment? We've always been guiding treatment. But now we are getting to the stage where treatment continuum has to depend on the lab. You can check for the term diagnostics. That's one of the greatest or new application of medical lab now, diagnostics, where patients are taking drugs and then you return back to the lab to begin to measure the effect of the drug. Now we also monitor. We monitor. That's when you come for HIV viral load, HBV viral load. You do BAR, ABL fusion test. We are monitoring. Then also, our function has left only answering one person's question. We are now answering a lot of people's questions. How do I mean? Our result is population oriented. When you identify that there is, this is Lassa virus, that Lassa virus does not belong to your lab alone. Immediately you get that result is useful for surveillance. So you make a noise about it. So we are now dealing with a population. Each of our diagnoses does not end with that patient alone. We are answering a greater community right now. Let's go. What is the sphere of diagnostics? I've talked about the landscape to show you that we now have a larger landscape that we are attending to. But now also our sphere of diagnostics has also changed. Before, we were just answering questions. Most of our questions that we were answering, they were based on cell and tissue. But you know that the organization of life starts from molecule down to cell, down to tissue, down to organ, down to organism, down to community, down to population. But right now, our sphere of diagnostics have really changed. How do I mean when I say it has changed? We are now diagnosing using organelle inside the cell. How do I mean? The mitochondria, as little as it is, has the capacity to contain some genes that has the capacity to be defected. And once it is defected, can lead to serious disease between a mother and a female child. So we have gone beyond the cell. Inside the cell now, we are now looking at the organelles in the cell. Our object of diagnosis before used to be cell and tissue, but right now our object has changed to DNA, it has changed to RNA, it has changed to proteins. Are we together? Little mutations that do not really matter before now matter to us. So our sphere of diagnostics have changed. We are using now the organelle, we are using now the cell. Before, most of our work is all about, is it present or is it absent for the cell? But right now we are checking if the cell is abnormal, we are checking if the cell is malignant, we are checking if the cell is infected, we are checking if the cell is responding to immune system. There's a lot that is happening in this field of diagnostics. Even for the organ, we are checking molecular subtype now. That's why when you are doing R2, EGFR is a common gene in cancer. But that you have the EGFR does not mean that this is what caused the cancer. It means that you trace the EGFR backward to be able to know what type of cancer is actually happening to the patient. Now, we don't also interpret our results based on an individual again. Our result is now one result in the midst of a population. Maybe I should give us an example so that we can come alive. It's quite unfortunate that I can't hear our responses, but let me be sure. Are we together? Can I have one or two persons respond?
Yes.
Okay. Thank you. Thank you so much. So we are not only interpreting results based on one individual alone. It's not that person alone. When you are doing paternity test, when you are doing paternity test, you are not interpreting that person's result based on that person alone. Let me give us a little expose. When we are doing paternity tests, we make use of markers called short tandem repeats or single nucleotide polymorphism. So if this particular person has that STR and the father also has that STR, you cannot just automatically say that that person is the father of the child because they both share the same STR frequency. You have to interpret that result and check it with the frequency of people in the population that used to have that STR. Let me say a particular STR. I'll just give a name. The name of the STR is XYZA. And the father had 15. The son also had 15. But in the population of Oyo State, 85% of people have 15 for that STR. You can't wake up and say that that person is the father of the child. So our result is not based on us alone again. We are expounding, answering our results based on a population. That is the concept of population genetics. I'm sure I'm making sense. So the sphere of diagnostics have really changed. It is bigger than what it used to be. It is far, far bigger now. And I also talked about the population here. Let me run. Now, what is molecular diagnostics? Molecular diagnostics is when you begin to diagnose a patient. Please, the word that I said diagnose is relative now, because I have told you that the landscape of diagnosis have really changed. So you could be predicting, you could be screening, you could be looking for viable organism. When you diagnose a patient based on the sphere of diagnosis that I mentioned earlier, which could be the organelle that contain the DNA, the RNA, you go down straight to probably the cell, the tissue, the population, anything that makes you make any diagnosis along that sphere is what is called molecular diagnostics. And I intentionally mentioned this so that we as medlab scientists, we enter into this together. Our samples for molecular diagnostics too has changed. It's different. In the hematology lab, you could have your whole blood in EDTA. In chemistry, you could harvest your serum and things like that. But here, whole blood is good for us. Plasma is good for us. Serum is good for us. Let me tell you, one of the things that we love so much in the molecular lab is the buffy coat. There's a lot of things that you can use that small, tiny buffy coat to diagnose. Because all the immune system, immune responses of the cell is in the buffy coat. So the buffy coat is a very, very important sample to us. We have what we also call the dry blood spot. You can use that to diagnose malaria. You can see also that we have swabs, different types of swab, all the bronchoalveolar lavage fluid, all those, that's our own sample. You can see urine, you can see stool. When we are doing microbiome study, you need the stool. You need microbiome study. When you are giving somebody a drug, that drug could be altering the microbiome of the individual in the gut. You need to study that. Because if you are giving the person the drug and the drug is going to reduce the microbiome, other disease conditions will creep in because you've already chased away the normal flora. That is now one of the tests that is being run in the molecular lab, where they are requested that a patient should go run a microbiome study. Okay, let's go. I want to rush a little bit now. We have different types of swab. We have the cells, we have the tissues, and then the cells. Let me mention one here. The molecular lab, when we're having, like, you are doing a human papillomavirus testing, we have different types of samples. But one of the samples that you need to be exposed to is the FFPE, the formalin fixed paraffin embedded tissue. That's a sample to us, a big sample, a lovely sample. So a lot of samples. You can see them over here. Also, you can see us collecting amniotic fluid, chorionic villus samples. That's what we use to detect people that are AS marry AS, and you want to be sure if you are not going to give her to SS, you need to give either your amniotic fluid, you need to give either your CVS sample, which will be collected, NIPT, that's non-invasive paternity testing. So there's just a whole lot. So we also test food and things like that. So let's move straight. What is the application of molecular diagnostics, one of the major issues that I found with molecular diagnostics that people don't really understand is most people believe that molecular diagnostics belong to medical microbiology. It's an error from the pit of hell. Molecular diagnostics belong to all fields of medical laboratory science. Every field that we belong has its own molecular end. Molecular itself is a field that applies to all. It's just that the most important, the one that have really caught our attention for the application of molecular has always been the medical microbiology, the infectious disease, the resistant genes, the outbreak tracing. But right now, hematology seems to be like they are the one enjoying it the most because there's a lot of cancer screening ongoing. They are doing tumor profiling, they are doing targeted therapy. Hematology is doing fusion genes, they are doing HLA typing, they are doing clonality test. It belongs to everybody. It also belongs to genetics and reproductive health. I'm sure we've heard of what we call the PGTA testing. The PGTA testing where you test a fetus if they are carrying some particular genes or whatsoever, that's molecular tests. It doesn't belong to anybody. It belongs to everybody. You could see also that the pharmacogenomics and clinical medicine, molecular diagnostics is doing a beautiful work over there. Also in public health and One Health, molecular diagnostics is doing everything. I say here, molecular diagnostics is not a niche test. It's a cross-cutting system for precision care and population protection. So let's move on. What is the central dogma? Our central dogma for molecular biology is DNA to RNA to protein to phenotype. I need to explain this. In the conventional or traditional laboratory, you don't get to see anything unless that thing has turned to the protein form. So you are saying that somebody is having an elevated amount of glucose, and you're saying this person is hyperglycemic. That person was not hyperglycemic yesterday. A lot has happened behind the scene before there is formation of protein. That protein is what you see as hyperglycemia. That protein is what you see as hypokalemia. That protein is what you see as a chronic myeloid leukemia. That protein is what you see as the antibody. There's a lot, but most of our diagnosis is at the protein level. But the molecular biology or molecular diagnostics draws you back that you can start diagnosing with the DNA. So when you diagnose with the DNA, we're able to detect mutations, we're able to identify chromosomal shifts, we're able to identify resistant genes, we're able to see traits that somebody is transferring from mother to child. That's DNA diagnosis. And that process, the DNA has to be copied by the RNA in a process called transcription. That means that somebody may have a disease, and that disease is not showing up because the disease has not been transcribed. So that means that we also need to establish a diagnosis at the transcription end, which is the RNA end, where we begin to measure gene expression. That is why somebody will have a disease today, the disease will continue to grow. The gene expression is increasing. That's what is happening. They said yesterday the WBC was 50,000, but as at this morning, the WBC is now 175,000. That means that there is an increased gene expression in the organ, tissue, cell that is producing those WBC. So gene expression itself is a test in the molecular diagnostic lab. And we do that kind of test in two ways. It could be absolute or it could be relative. Don't let me bore you with that. I believe that as you come closer to molecular, you begin to get used to all those things. But sometimes when you are doing your viral load, a viral load is an absolute molecular test. Tells you the amount of a virus present. So, and it's an example of gene expression test. So I leave that. So also, we also do protein diagnosis. That is when we begin to check for biomarkers, begin to check for host immune responses. That is proteomics. So when you say it's DNA that you're working on, that is genomics. When it is RNA, it's transcriptomics. When it is protein, it is proteomics. So we can detect along this central dogma. We can now detect disease along this central dogma. That makes it very beautiful. So what are the things you can detect using DNA? You can be able to find out if somebody is carrying a mutation, inherited or acquired variant. You can, when you want to do paternity, it is DNA that you need. That it is why it is called DNA paternity test. But that's not the name. We are all med lab scientists, so let me quickly correct that notion. It is not DNA paternity test. It is called relationship testing. It is when it is between a father and a child that it is paternity. If it is between the mother and the child, that is maternity. If it is between siblings, that is siblage tests. If it is between a grandparent, that's grandparental tests. So many forms of the test. Also, DNA can help you to identify microbes, viruses. It can help you to identify if a woman is carrying a mutation in her mitochondria. It's very, very important. And that will also lead me to something else. There are different types of DNA. DNA. There are plenty. We have what we call genomic DNA. We have what we call the microbial DNA or viral DNA. We have the mitochondrial DNA. The mitochondrion itself has a DNA. Plasmid in bacteria has its own DNA. Another thing that is now trending now that we are using to diagnose is what we call the cell-free DNA. This has been very, very important in prenatal testing. You harvest cell-free DNA and then diagnose with it. We have what we call the circulating tumor DNA. You have what we call the methylated DNA. We have the 16S RNA gene DNA. All of these that I'm talking about, when you have a sample that comes into the lab, you must know the type of DNA that you want to extract. Because if you don't know the right DNA to extract, you won't get the right result. There are also types of RNA. Let's see. We have viral RNA, we have messenger RNA. When we are interested in gene expression, we focus more on messenger RNA, but that's not the only type of RNA. We have what we call the fusion transcript RNA. When you are doing leukemia tests using molecular method, it is fusion transcript RNA that we are working with. We also have ribosomal RNA, which is also diagnostics. We have microRNA. In the next few years, in about 5 years, this microRNA will take over all the spaces that everybody has covered before, because there's a lot of signature in microRNA. We have what we also call the long coding RNA. We have the circular RNA. We have the cDNA, but cDNA is not diagnostic. Please, it's not diagnostics. It's not diagnostic. So let's move. I may skip this. Molecular tests will help you to detect and identify, help you to quantify and monitor, help you to characterize and classify, help you to predict and personalize. Molecular tests will help you to establish relationships. Molecular tests will help you to protect population. So let me stop here before going. At this level, I'm sure we got me to this level because I want to go a little bit deeper. Did I make sense to this level to anyone? Please, can I get responses? I can't see your chats, but I can hear your voice. Oh, am I disconnected? Hello? Hello?
I've been following you, sir.
I think I'm disconnected.
Yes, we can.
Okay. Sorry, I was worried that I couldn't hear anybody respond to me. So, okay, let's move forward. We've talked about everything you need to know about molecular diagnostics. So what are the types of molecular diagnostics that you are going to come in contact with? I would divide them into three. Number one is we have PCR diagnostics, we have sequencing diagnostics, and then we have CRISPR diagnostics. Number one, PCR diagnostics. This is when you have to do detection, prevention, anything that molecular can do, but you can do it using the PCR, which is called the polymerase chain reaction. And I have brought it here that we have what we call the conventional PCR. You can see it on your slide. All the things we learned in school, denaturation, annealing, extension, they work. That is the concept through which PCR work. If somebody is suffering from a disease condition, let me name a disease condition that I love so much. Let's say, we want to know if this person has multiple myeloma. This person has multiple myeloma. We look at the sequence of multiple myeloma. You look through it and look for places that do not change. Those places that do not change, those are the places that we call the conserved regions of the gene. The conserved region, they don't change. So you design a primer. A primer is like, I'm sure most of us have, you probably grew up around the Badr or something like that. And you know that if you are fetching water, most people now, they don't fetch water, but if you are fetching water and your fetcher, Doro, falls inside water, you need something to bring it out of the of the water. You use something that they used to call oloto. You call it oloto. Once you put it inside, it will go and touch one part of the so-called duru and drag it out. That thing is like the oluto, that is like a primer. So you have found the conserved region that do not change in the genome. You send in something like that, which is a primer, to go and drag it out. And once you're able to drag it out, you have been able to detect. So you design a primer that is specific for multiple myeloma. Once you are able to design that primer and then you run a PCR reaction, denaturation and Snellen extension, and the thing becomes positive, it means that person has multiple myeloma. So that's the concept through which we are using to do it. And after you are done doing it, you need to run a gel electrophoresis. That's what you are seeing behind. That thing that has so many markers like line. That is gel electrophoresis. That very long line is what we call the DNA ladder. And then each of the remaining has, you are seeing the DNA bands. So it helps us to know if there is presence or absence of a disease condition, pathogen or whatsoever. So, or a gene or something. Another thing is we have what we call the qPCR. The qPCR is different from the conventional PCR. The qPCR helps you to measure. How do I mean? The conventional PCR helps you to detect the presence or absence, but the qPCR helps you to measure. It gives you real-time information. So you are saying this person has HIV. Conventional PCR says this person has HIV-1. qPCR is saying the person has HIV-1, but the person has 1001 IU per microliter of HIV-1. You see, you can see the difference. That is real-time PCR. It helps you to measure. It helps you to measure the exact amount of a pathogen, disease, gene present. I'm using this not only pathogen alone. There's something we call gene copy number. There's something we call copy number variation, CNVs. qPCR will do it for you to help you know the presence of one gene and the number of that gene that is present in your body. qPCR will tell you. Beyond that, now we now have what we call the digital PCR or droplet digital PCR, ddPCR. Now, before we were measuring everything as a whole volume, but the ddPCR is different. It begins to measure that your fluid that was collected. When we collect blood fluid and put it inside the pipette and you are dispensing, the ddPCR will measure it droplet by droplet. It will check it one cell after the other. So any result you have from droplet PCR, believe it, it is true. It is true. So let's move to the next one. I've talked about PCR techniques as a form of molecular diagnostics. The next technique that I'm talking about is sequencing technique. So let me just move to the next. I've mentioned this already, so I wouldn't want to bother myself about this. Now, let me say something before I go into sequencing techniques. PCR has given us the capacity to do something, the capacity to detect many things in one single reaction. That reaction is what we call the multiplex PCR reaction. That means that you can look for about 15 diseases and say, people who suffer from sexually transmitted disease, if it doesn't have gonorrhea, we have syphilis. If it doesn't have syphilis, we have Epstein-Barr virus. If it doesn't have Epstein-Barr virus, we have human papilloma virus. You can pull all of those tests together and put it in one single test. That is multiplex PCR. So when you run that one single test, you will know if the person is carrying HSV, the person is carrying HPV. Most times, imagine if you are running HSV, you run HSV alone and the person is positive for HSV1. You say the person has HSV. But from experience, using multiplex PCR, we have found out that people who have STI most times used to carry more than one type of STI in their body. People who have HPV sometimes used to carry more than one type of HPV in their body. I'm sure it makes sense. So that is multiplex PCR. So let's move on. Let's move on now. Now we are talking about sequencing diagnostics. Sequencing has been one of the things that answer our question the most. It has answered our questions a lot. Because most diseases that we encounter in the lab that they come for screening for, they are diseases that happened because of mutation. Especially most chemistry tests that becomes elevated is something that is mutated. So even though they monitor it today and give the person therapy, the thing will likely reoccur again because it's a genetic problem. So you can only use PCR to identify what you have known that exists. That's the word. I know that there can be this particular gene causing this thing. Go and run PCR. But what if you don't even know what is causing what you are interested in? You don't even know. In that kind of case, they say, we don't know what is wrong with this person. They'll give out the drug. Just take the person's sample and go and sequence it. Once you're able to sequence it, you will see so many DNA, you see so many RNA, and then from there you can have an idea of what is happening. Or example, you feel like, oh, this person is having liver problem. And you say, this liver problem, you can say, okay, the most important genes in the liver, just target 2 or 3 of them and go and sequence that gene. You target the gene using PCR, get out the result from the PCR, then do sequencing. That is what we call targeted sequencing. And you begin to look inside the sequence. Is there something different? You may be shocked. Immediately, you just find out that where there's supposed to be T, it is A that is there. And you know that 3 nucleotide sequence form 1 protein. So somewhere that is supposed to be CAT became CGT. It could lead to the formation of another protein. And that protein may lead to a lot of problems. So the problem that you are dealing with may be from that one single nucleotide change. And this type of sequencing process is what we call targeted sequencing. And that is done majorly by Sanger sequencing. Sanger is just the name of the man, is the person who identified it. And you have heard Sanger sequencer. Sanger sequencer is just a machine that does Sanger sequencing for you for targeted sequencing. But life done beyond that too. Sometimes you will do targeted sequencing and you still don't have your answer. So you may have to do what is called whole genome sequencing. You have to sequence the whole genome of an individual, sequence the whole genome of a plant, sequence the whole genome of a bacteria or of a virus. That's how we identify outbreaks nowadays. That's how they identified Ebola when it came. They did sequencing, identified it, and they begin to do bioinformatics analysis. I need to rush because I have a lot to talk about. So, whole genome sequencing, it helps you to identify. Another one is that you may not be interested in the whole genome of the organism. The genome is made up of the exome and the intron. The exome is the coding part of the organism. That's what we say, but that's not true, but that is what is believed. The exome is the coding part, so you may just focus on the functional parts. That's the word. So you can do whole exome sequencing, but there are many types of NGS machine. We have the PacBio, we have there are a lot. And then we have moved from that again. We have what we call the third generation sequencing. That is the third generation sequencing. And this is the kind of sequencing that allows you to sequence without even culturing. You don't need to culture. And we have the example of that is nanopore sequencing. The reason why I'm saying all of this, my story, is that in Ibadan alone, there are 4 sequencers in Ibadan. Let me use the word 4 sequencing capabilities in Ibadan. I'm still going there. So let's go. What is now the diagnostic technology looking like now? Amplification using PCR. We have next generation sequencing. We have what we call the NI non-invasive profiling, where you use the CF diagnosis. And then we have the higher resolution like the dPCR and the ddPCR. One that I missed out here that I'm not happy that I missed out is the CRISPR. Most people know CRISPR as a tool that it is used to edit a gene and then affect a clinical outcome. CRISPR is more than that, sir. CRISPR, there's a part of CRISPR now that you can read on later. For those who don't know, there's something we call DCAS. CRISPR CRISPR. Now, people are now using the knowledge of guide RNA. The major thing in CRISPR is called guide RNA. Guide RNA can guide you to a particular location to locate something. So if you want to remove one particular nucleotide and remove one particular gene and put another gene there, or you want to knock it off or something, you use guide RNA. People are now using that guide RNA again to now design diagnostic tools. That's where molecular is actually going. Because we in Africa, the outside world do not really know our problem. They are doing the solutions based on their own problems, not on ours. So now we also can now know what is our problem, design a DCAS that will target our problem. And then that DCAS, you can use it in a PCR machine. You can use it even in an ordinary bedside point of care testing kit. So DCAS is the in thing now. Thank you. So let's move. I've talked about a multiplexing system where only one sample can do so many things. So I won't bother my head on that. Now, why did I tell us all of this story? WHO this year did a conference and they spoke on precision medicine. And they gave a verdict that precision medicine is when you now use health data to do prevention, diagnosis, and treatment while accounting for individual variability. They now say that it's important that we always get clinical data. They said after you have gotten clinical data, it's also very important that you get molecular data, get genomic data, know the individual variability, then tailor your decision. This is now the new way to diagnose people in the world, according to WHO. You know, we are in Nigeria now. How now? It takes time before it gets to us. And then you also monitor the response. So let's go. I will leave all of this. You can see it when you have the slide. I think I'm going to leave all of this also. You can see it when you have the slide. Now, in response to that, because WHO has been saying that for a long time, but now Nigeria also had their own meeting. They call it the National Genomic Surveillance Meeting in 2024. And in that meeting, they were able to unify how precision medicine is going to be in Nigeria. And one of the things is that Nigeria is receptive to the idea of precision medicine. They are receptive. So they are gradually integrating it into our healthcare system. It's not profound, but they are doing it. As at 2024, Nigeria has over 24 sequencing capability location. I'm telling you, I'm not saying the machines, I'm saying location. That we have 24 locations as at 2024 that Nigeria has sequencing capacity so that if there's any outbreak, they can go there and know what has really caused the outbreak. We are growing. Now, let me shock you. In each of those locations, I've been to run. That is SGID. SGID has over 9 sequencers. UI has a minimum of 4 sequencers. So, the integration part of the National Genomic Surveillance has increased. I think about a month or two ago in Lagos, Nigeria created what we call the National Genomics Center for multiomics. You can Google it for multiomics. So there's a building now in Lagos where Nigeria has different sequencers in it. Who are the people who are doing that place? I can assure you they are not med lab scientists. And it ought not to be. So Nigeria is really, really creating for national genomic surveillance. Nigeria is becoming prepared for it. But my question to you is, are you prepared for it? Are we as med lab scientists preparing ourselves to occupy those spaces? I'm sorry that I'm already doing a lot of things that people may not like, but it's just the truth. And one of the motto that came back from that national genomics meeting was that everybody is involved in genomics. Whether you are the local government or you are the tertiary hospital or the reference center, everybody is involved in molecular testing. At the local level, people are now being trained for correct sample collection, labeling, storage, and referral. See, if you collect a sample and you don't store it right, the whole thing has spoiled. So it's an hub. It's an hub. Nigeria is doing a lot around that hub. It's a tiered system. Let me run quickly. Now, I will end that part before I go to bioinformatics on the UHC, the Universal Health Coverage, that has started to trend for about 3 years now, which is what has been decided globally, that in the whole of the world, we must practice an all-inclusive healthcare system where primary facility play a role for secondary facility play a role for reference center, and then whatsoever comes from reference center gives a public health answer. You can study this slide to understand what I'm trying to say. So I will also move this. We should know that all our labs should be compliant with ISO 15189, but I will leave all of that. Oh, sorry. I'll come back to this slide. I really want to talk about bioinformatics. Now, I have talked so much of molecular diagnostics. I will not leave without talking about bioinformatics, because molecular diagnostics will generate data. Bioinformatics will make sense of that data. I was downloading a bacterial genome. Okay, by the way, we also have our own sequencer now in my own university here in Babcock. We have 2. Now, I was working on a bacteria that I was working on. I needed to download the bacteria. You can't imagine that bacteria, after I downloaded the work that I wanted to do, was about 60 something gig. Now, 60 something gig of ATTG, AGG, AM, GAGG, that is just alphabet. You need to make sense out of it. And if you need to make sense out of it, you need bioinformatics. Bioinformatics simply means biological information. How you extract biological information. So molecular diagnostics will produce the biological data. Bioinformatics will give you the biological information. For example, if you are using qPCR, it will give you CT. That's what is going to be giving you, CT values. When we're doing COVID, you remember, somebody will just say, the CT is 35.25. That is CT value. That's not a diagnostic information. You have to use bioinformatics to interpret it to get to give you a diagnostic information. We have curves. We have copy numbers. Those are the things that are coming out from the molecular lab. We have focused genes. We have deep coverage. Those are the things that are coming out. Somatic variant. That's our own result. Those are the things that we're talking about. You see, after we have analyzed the sample, we found out that so genes are highly expressed. You will need bioinformatics to be able to interpret if they are highly expressed or not. Now, the interaction between molecular diagnostics and bioinformatics is interrelated. How do I mean? Bioinformatics give molecular diagnostics the information. Molecular diagnostics works on the information and return the information as data back to molecular diagnostics, back to bioinformatics. That means that it's a circle. And that will lead me to the concept of what we call the dry lab and the wet lab. The dry lab is a sweet lab. It's a lab that everything we do there is dry. Let me use that word. How do I mean? There is no pipette. There is no pipetting tips. There's no solution. Everything that you see in the dry lab, all the things you have in the wet lab, they are coded in the dry lab. How do I mean? You can check if the primer is working in the dry lab. The primer, you said, I told you the other time that there's a particular disease, multiple myeloma. You design the primer towards it, and then you now come to the lab to run it. Before you do that, better don't do that. You have to do dry lab first to check if the primer, you use dry lab to design the primer, you use dry lab to test the primer, if the primer is going to work. So after you have tested it in the dry lab and it works, you come back to the wet lab to practicalize it. In fact, there is nothing that we've been doing in medical laboratory science that did not originate with dry lab. Dry lab is the part, all the reagents that we are using, they were dry lab tested. How do I mean? They were dry lab designed. Okay, this thing is going to detect HIV-1. They used dry lab to isolate HIV-1. They tested it on the computer. Another name for dry lab is what we call in silico analysis. In silico analysis. So after they found it that it's going to work on the computer, they will now sell it. You in the wet lab will now be using it and say, it is working. It is working because it was dry lab that created it. But in the case of molecular diagnostics, dry lab created it, sent it to the wet lab. The data is generated in the wet lab and returned back to the dry lab for analysis. Can I just 1 minute of 30 seconds of rest just to be sure that I am making sense? Any response, sir? Am I still on track? Are people still hearing me?
Hello, sir. Your slide. We are trying to figure out where you are exactly, but I don't know which page are you, because we are on the future frontier of precision diagnostics. We don't get.
No, okay. Are we not using the same one?
Sir?
Okay, can you see where I am now?
No, not yet. Not at all.
Can you see this national genomic surveillance?
National genomic surveillance. Okay, sir. We are going there now.
Okay, so you are going there, right?
Yes. National genomic
surveillance. So go one step lower. Yeah. Are you here now?
Yes.
I talked about this, that things have really changed. The new tiered network is that from local lab down to reference center, everybody must be involved in healthcare. And you can see what I call the universal health coverage. Ah, and you didn't tell me, sir. Are you seen this quality assurance and ethics? Are you there, sir?
Yes. The third network.
Just keep going one slide down. Okay, one more slide. Okay, this is this place.
Okay, before.
Where I come down again. Are you there now?
Okay.
One more step. Go down one more step.
One more step.
Quality assurance and ethics.
Forward. So,
are you there?
Yeah. We are going forward, right?
Yes, just go one more step down forward.
1, 2, 3, 4. Yeah, that's it, sir.
Okay, let's go. Have you seen the evolving laboratory professional?
Yes.
Okay.
Yes.
I'm still going down there, but I'm not talking about it now. So come here, can you see this molecular data? Come to where this molecular data is.
Yeah. Yeah.
Are you there?
Yes. Molecular data to reliable decisions.
One more step, please.
Okay.
This was where I was talking about all the kinds of data that you can use for that comes out of the molecular lab, the CT numbers, the curves, the genes, the somatic variants.
The curve, CT, copy number, exactly.
Yeah.
And the targeted
WS. Wow, I didn't know we've missed each other a lot. Nobody told me. I'm really, really sorry about that. So now I'm going to the next slide. So let me take this slide and then I'll go back to the next slide. I was saying that the dry lab, I'm sure we all understood what I was talking about, dry lab, wet lab, when I did that.
Yeah, that is even where we couldn't get to.
Yes, we do.
Oh, sorry. What happened is that slide does not exist. I'm just the one talking about it.
Okay, thank you.
Thank you. So let's go now one more step. So how do dry lab and wet lab enter each other? That's what I want to talk about now. That's what I want to talk about. There's a clinical question. Somebody is feeling, the person is having malaria. They say he's having malaria or the temperature is increased or is having fever. That's a clinical question. After they've done a clinical question, a sample will be collected. The metadata too will be collected. Then after that, you check the quality control the samples that comes into the lab. In the molecular lab, we do a lot of quality controls. Now, let me tell you, when we are running our samples in the molecular lab, there are about 5 different types of control. The first one is called the non-template control. Non-template control. It doesn't contain anything. The next one is what we call the negative extraction control. Because you need to extract the DNA before you amplify. So you need a negative extraction control. You need a positive extraction control. You need a negative amplification control. You need a positive amplification control. After that, we have standards and then we have controls. You see that we do a lot of controls in the molecular lab. But after we have done all of that, any result that we have, we will analyze it. We will analyze it. If it's a sequence result, you do what we call sequence assembly. If it's in a count, you can do what is called count. You do annotation. Then after you've done annotation, then you interpret. Then after that, you report. If you didn't get it, don't worry. I'm still going to talk about it somewhere around here. I'm using something close so I can see. I talked about targeted sequencing the other time. Somebody is having breast cancer and they are thinking, oh, this breast cancer, it could be less. Have you screened for BRCA1, BRCA2? I'm sure we've heard all these terms. They said go and screen for BRCA1, BRCA2. What we used to do is very simple. The patient comes into the lab, the sample is collected, you extract DNA from the sample. After you have extracted DNA from the sample, you create a primer for BRCA1, BRCA2 and target it. Once you target it in the patient and you have it and you find out that the patient has BRCA1, BRCA2, the next thing is that, this person has BRCA1, BRCA2, but does this person has mutation of BRCA1, BRCA2? Because it's that mutation itself. The mutation itself is highly cancerous. So you now do mutation. You need to do what's called targeted sequencing. You send the targeted BRCA1, BRCA2 for sequencer using Sanger sequencer. Now, when you have the result will come back to you as either faster. Go to the other side now. You can see. Don't leave the slide. Just the fast queue. The result will come as something like this, either faster or fast queue. The first thing is once you have the result, you have to do QC. This is all on computer. you will run QC on computer to check if the sequence makes sense or it's a fake sequence or it contains contaminant. You know, in the lab, you can't see contaminant unless it grows on plate. In the in silico lab, we can see contaminant. I don't want to bore you too much on our terms, but there's something called check M. With check M, you can be able to know the amount of contaminant in a sample. The next thing is you align it. The word align is what you people have called blasts. You already have the value already. You have the sequences. You need to blast it. Who is this sequence looking like in the whole of the world? Once you blast that target, it must give you BRCA1, BRCA2 again and give you the exact mutant. Do we get what I'm trying to say? MLS Temilolu, are you there, sir? MLS Temi. Oh my God. Okay. Let me just assume that I'm being listened to. So you blast. It is when you blast that you identify.
Can you hear me?
Yes, sir. Can you hear me now, sir?
Yes.
You are
We
with
are
me,
following.
Abi?
We are
Okay.
following him.
I don't want to lose anybody.
Not
So
at all.
once you have the FASTA or FASTQ file, the next thing is that you will align. You will align, then you will blast. You do blasting and then you align. When you blast, you're able to say, oh, this thing is BRCA1, BRCA2. So that is when you blasted. You now align what you have blasted with the reference. That's why we always have reference. After you have done it, you check it with the reference. You just find out that maybe one of the nucleotide sequence is what is not correct. That is when you do what is called calling, base calling. You call. I say, I have found out it is nucleotide number 35 that is not the same as the reference. Assuming that nucleotide number 35 is T, the reference is T. If it is T, it would have been normal. But as of today, it is A. That means that it is a mutant. Then after that, you now annotate. We have our own special website for annotation, which is called clinical variant or ClinVac. You can look at it. Not every mutation in this life makes sense. It's ClinVac that will let you know if the mutation makes sense or does not make sense. Then after that, you generate your result. So let me just, I've talked about all of this. So I will rush all of this. I don't want to waste our time. So you can look at it very well. I'm coming. Okay, are you moving with me, please? Let me go from where I left. This is one slide lower, another slide lower, third slide lower. Okay, let me stay on this slide. Stay on this slide, please. Nowadays, you don't need to sequence a whole bacteria before you're able to identify the type of bacteria that it is. Now, you can just, once you just extract the DNA, you can run what we call the 16S RNA, the 16S RNA analysis. If you're able to do the 16S RNA analysis, you can be able to identify all the resistant genes that is present, all the virulence gene, all the plasmid, all the mutation, and you wouldn't have even cultured. You don't need to have culture. After you have done all of that now, using bioinformatics, you can now draw the phylogeny. How do I mean? You want to draw what is called the source tree. You want to know the source. Oh, this bacteria that we identified. There's a case. Let me mention the particular case. I won't tell you the name of the hospital, but there's a particular case where children were dying in the emergency ward. Children were dying in the neonatal ward. And it is just one strain of bacteria that was disturbing them. I'm coming, please. It's just one strain of bacteria that was disturbing them. They have been isolating the bacteria, but they don't know if it is the same bacteria. You need bioinformatics to answer that question. Because after you have sequenced that bacteria and you sequence others, you are able to see if they belong to the same clade. You belong to the same family. They may be the same bacteria, but they do not have the same virulence genes. They do not have the same resistance gene. They don't have the same resistance pattern. Also, when you do all of that, there's something we call typing. There's something we call typing. If you type a bacteria and you find out that it belongs to a very high MLST typing, run away. That can create an outbreak. So let's go to the next slide, please. I really want to run. So all the analysis that we have in the lab, all of them goes back to bioinformatics. You cannot take away bioinformatics away from those analyses. So this is the next slide after the former slide, and this is the next slide, the pipeline tools and database for bioinformatics. I mentioned the other time that if you want to do QC, we use fast QC, we use fast P, we have multi QC. But when it is time for us to do analysis, we do what we call the spate, the fly, the BWA. When you want to know a variant, you want to know, is this person, is it the variant? Is it the normal variant? In molecular biology, when something is normal, we call it wild type. When something is normal, I don't understand who named it. But when something is normal in molecular biology, we call it wild type. When it is abnormal, we call it variant. So for you to do variant, you need tools like GAT-TK, and then for you to interpret, you need tools like clinical variants. And then that's it. So this is the future of precision medicine. This is how precision medicine is going to look like in the future. I'm already reaching the end of my slide. This is what precision medicine will look like in the future. You can see that we have what we call next gen portability. There'll be machines that can give you results almost. Our machine, which is called the nanopore sequencer, can give you results from as early as 2 hours when you sequence a sample and you know what the sample is. So you always take those results. In the future, we will not have to be interpreting those results by ourselves again, because AI and machine learning will be helping us to interpret it. Also in the future, expect when people will do a lot of multi-tiered tests, where people will do genomic tests for a particular patient, do transcriptomics tests for that patient, still do proteomic tests for that one single patient. Because if you do these three, it will give you an awesome view of what the patient is going through. And then lastly, expect CRISPR to dominate the market. Please, I cannot overemphasize this. DCAS is here. DCAS is here to reign. CRISPR diagnostics, DCAS, will be the new way to detect pathogens, to detect diseases. So who should you be? The new molecular diagnostics, the new medical laboratory scientist. I'll go back. That slide is somewhere here. I will really show you. I'm interested in this slide. Sorry. So as a new medical laboratory scientist, the evolving laboratory professional, this is who you have to be. You have to be molecularly literate. You must know molecular diagnostics. Don't run away from molecular diagnostics. Don't run away from it. You have to know it. Because at which tier that you are, whether you are at local tier, you are at secondary tier, you are at tertiary tier, you are at reference diagnostics. So you need to understand the genomics, understand multiomics. You must learn also data interpretation. You must know bioinformatics. I bet to say, all the trainings you see online where people are talking about bioinformatics, that's not your own bioinformatics. So, because they are talking about general bioinformatics, you need clinical bioinformatics or diagnostic bioinformatics. They are totally different from each other. Clinical bioinformatics, our major tools are around CleanVac. You can write it if you've never heard of it. Clinical vac, CleanVac. We do a lot of CleanVac, we do a lot of GAT-TK. They are totally different. Next thing is that as the new generation med lab scientist, you must have a cross-disciplinary outlook. You must have a cross-disciplinary outlook. You must be able to cooperate with clinicians, because these days now, clinicians are there, epidemiologists are also with you, bioinformaticians are there. I always tell myself, med lab scientists, we have to become the epidemiologist. We have to become bioinformatician. Nobody can understand your data more than you. It's not possible. But it's quite unfortunate we are still not filling all those spaces. And then also, we must also give leadership. It's very, very important. We must give leadership. The new universal health coverage is saying one thing. The world has moved into molecular diagnostics. It's either you lead or you are being led. So I will end my talk this morning like this. Medical laboratory science. Will you lead this revolution, or will you be led by this revolution? I want to say thank you for having me on this platform. I've enjoyed myself, and I'm sure you have also enjoyed yourself. So I would like to listen to feedback, questions, and things like that right now. I have suffered for about 1 hour because I couldn't see you. Now I want to see you. Thank you.
Thank you so much. We really enjoy the rich presentation. Thank you.
All right. Thank you so much. There is a poll that is on the group that has been shared. I hope everybody is seeing the poll right now. So we want to see what your feedback is on that poll. So start. It looks like a poll, but the best answer will be declared at the end of the meeting. And thank you so much, Amlsn. That was a wonderful presentation. I know when we are going into molecular diagnostics world, it could take you years before you can even say you've had a true mastery where it is essential. However, for you to be able to take a work of over 10 years experience into short minutes, it's amazing. And not only that, you even dive into the futuristic relevance of the molecular diagnosis and molecular techniques is exceptional. So thank you so much. And I think we should give room for question and answer for just like 6 minutes. 6 minutes that we have. So if we have anybody with questions, you can just let's see, do we have a question and answer session? Okay, just drop it in chat. So we are going to acknowledge the question. So if you have a question, okay, somebody has a question. There's a question already. Let me put it on stage. So yes. Can you see that question, sir? It's on stage
Yes,
already.
I can see the question,
Okay,
sir.
that's
The
one.
question
So if we have
says,
any other one, bring it to the stage.
okay, thank you, sir. The question says, I work on Giardia lamblia, genetic variation of Giardia lamblia among selected primary school pupils. There was a time my supervisor informed me to send my findings to GenBank. What does that mean? So I think I need to just explain that we are all working from, let me use the word, the world is now a global village. And there are some tools that connect all of us together. One of those kind of tools in genomic medicine is what we call the National Center for Biotechnological Information or Biotechnology Information, NCBI. And one of the channels on NCBI is what we call the gene bank. What you do there is that when you do a work that you probably sequenced or you got something new, you are expected to deposit it on gene bank. Once you deposit it on gene bank, it helps to inform another person that, oh, a variant of Giardia lamblia now exists. And they may want to design a diagnostic kit. Somebody is saying, okay, the one that you have discovered in Ibadan, the Giardia lamblia kit that we have before is for Africa. But when it captured the one for Africa, it doesn't capture that type of Giardia lamblia in Ibadan. But you have done a work on Giardia lamblia in Ibadan. Once you deposit it on gene bank, it becomes visible for everybody to see. And anybody who wants to now design a diagnostic kit can now design a diagnostic kit based on that your own Ibadan Giardia lamblia. That means that next time when people go to the lab to run an analysis on Giardia lamblia, they will just use the Ibadan Giardia lamblia gene that has already been deposited on gene bank. So that's it. It's very, very important that you deposit because you are adding to the body of knowledge. I beg of you, if you have not done that depositing, please deposit it. It will be deposited in your name. And anywhere in the world where they are making use of that Giardia lamblia, they will be referring that you are the one who deposited it. Are we together? Thank you so much. Oh, another question. He said, what will you say is the difference between dry lab and wet lab? Just like I said the other time, the smallest way to explain it is that the lab where your hand is wet is the wet lab. As long as your hand is wet, you are mixing this with this one, mixing this with this one, mixing this one. That is the wet lab. But the wet lab is not the wet lab is a diagnostic-centric lab. The dry lab is an innovative-centric lab. The meaning is that the dry lab informs what goes on in the wet lab. You can't say that there is a kit that we are using today that came from heaven. None of them fell from heaven. They came from the dry lab, where people had to work on sequence that we've already sequenced before. They worked on it and use the sequence. They use the sequence to answer a particular question that can help us to continue to diagnose. Let me give you an example. Somebody discovers schistosomiasis, sequence the schistosomiasis. Nobody have been able to use a kit to detect schistosomiasis, but you found schistosomiasis in the urine, sequenced it, deposited it online. The dry lab, somebody will now take that sequence of the schistosomiasis and use it to go and design a kit and send that kit back to the wet lab. So I'm sure I've answered that question now. So they go hand in hand, but everything about the dry lab happens in the computer. We are computer-based people. And then wet lab is when you begin to get your hands wet. Get your hands wet. So I think I've answered that question.
Right. Where did I see the two?
You saw them here, sir.
All right, thank you so much. Should I address you as a prof and doctor already? Because I can see the way you are dissecting this as a professional. You went far deeper. I don't know what else one could say because you really have a deep experience and understanding of what you're teaching. So we see something. I just love the fact. I just want to add this in simple word also in addition to the wet and dry. I don't think I have much time, but let me just say it in a simple sentence. When you are thinking of the wet, think of it in the experimental part. I just want to add that. And if I'm wrong, Prof, you can also correct me, or Prof in the meeting. So the second part is the dry. When you're thinking of the data behind everything you were doing, think of the dry. So that is just the basic separation between them. So when you want to go into experiment, know that you are going into the wet. When you are going to information about what you are doing, go to the dry. So that is, and you are not touching anything like you explained. I hope that simplifies it for that person.
Yes, sir. Thank you, sir.
All right. And I think I'm in sync with you, sir. Thank you so much, sir.
Perfectly well, my boss.
Thank you so much,
you,
sir. So, I have this thing I always to say to my people whenever we are having this kind of session. I will say, we always want to. There's a Yoruba adage that says, "We love." There's a Yoruba adage that says, "Owuni." So, and I love using in the English. The English is, you always prefer to, you want to eat the meat for so long in your mouth, but the drawer of the throat will not let it stay there. So you just see it sinking in directly. So that's exactly where I'm talking about that. This has been an exciting session, and it's something that we would have loved to, gain and keep delving more. But thank God we have the slides. We also have the recordings available for those interested. And if you would love to also get hands-on experience, I'm glad our facilitator is also ready to work with us. Just reach out to the team, the Oyo State Hospital Management Board, Association of Medical Laboratory Scientists of Nigeria / the State Ministry of Health. You will get a right contact and we'll be able to give you well detailed. And I know that there are some other programs that you might not be able to make an announcement of, but be on a lookout. You will get more hands-on experience on programs like this. So thank you so much, sir. I will love to give rooms to the chairman of the committee to give a thank you note. Thank you.
MLS committee.
Thank you, boss.
As I call my friend, I call him Prof. Committee. So this your presentation, in fact, is just a testament to who you are and that your choice was never a mistake. So we really appreciate you. We appreciate that you share the slides with us so that we can share with our participants. And if we still need any, possibly like the moderator said, maybe we have some people that are, planning to make a career in the field, and they might need a kind of, tutelage. I will still reach out to you, and we hope that, though I know you are very busy, but that you will be magnanimous enough to create time for us for such a, guide. So once again, thank you so much. We really appreciate your presentation. Thanks.
Thank you, sir. It's a privilege also. Thank you so much. All these people, they are my oga, they are my friends. So I appreciate the privilege. Thank you so much. Have a wonderful conference. Thank you.
Okay. Thank you so much. The email for enquiries and further contacts will be dropped for us, for anybody that feel like giving and more inquiries or making more inquiries rather. So you can just make it and send it there. Then for the poll, I don't know, maybe many of us have been attempting the poll that is on the group. It's just like a brief assessment of what, at the onset of the meeting, we tried to have a pre-test or pre-assessment, and we saw that many have a little or no knowledge. And there is a question that is there, which just synchronized what was asked, that for molecular diagnosis, what is the role of bioinformatics to it? It's a very simple statement that was said by the facilitator while he was speaking. You are already given it, and we also cite the reference some answers there on the platform. So pick any answers you feel like is your best option, just select it in the poll. I think the poll is visible for people to select, so you should be able to click and let's see your answers. At the end, I know that the facilitator himself already seen the right answer on the question. So it's quite technical sort of, but if you look at it in details, it's just to capture that truly you understand. So now we are moving to the next session, where we are going to be engaging on the medical legal. Do we have the facilitator on ground already? I've not yet seen the facilitator. He's not yet on ground. So while we're waiting for the facilitator, I think we can just have let me check the poll and see maybe we've been able to go on. So yeah, so that is the email to reach out to. You can see it's moving on the screen. So that's the email if you want to send us an email and you want us to follow through or any inquiry, please reach out via that email. Okay. Yes, it's the poll. So if you have already selected on the poll, we will be able to read. We should be able to read by now what the answer has been. Can anybody be able to look at the poll properly? Okay, somebody said they can't see the poll. Okay, somebody said he's having an internship. That's beautiful. So it's just going to be our meeting. The redeemable CDP points. I just finished internship. Oh, there are a lot of questions. Yes, the recording will be available. Yes, any interested MLS can be trained and mentored by the professional. Just reach out via that email and send it to us. So just reach out. Any other thing? Any other question? Converting this training to a CPD points we are not certain about that. So you might have to contact the council on it. But the recording and evidence of training will be made available, and even the certification will be given to those who participated in attendance. So any other thing? I can't see the poll. Okay, so we were told that they cannot see the poll. So maybe let me take the poll down, or let me reset it and see if it will come. Okay. Have you seen it? Okay, so Alba now. Okay. Okay. It's there. It's there. So we are the next facilitator is having some technological or technical difficulty at the moment. So I know he's going to join us very soon. So please let's exercise some patience. Or a patient, or is it a patient or patient? Sorry, sometimes I need to relearn and unlearn my English. Okay, so please exercise patience. Okay, but can we all see the poll now? Or is it still not? Okay, they say I can't see the poll. So can anybody see the poll? Is it still there? So we're waiting for you. Okay, somebody said no. Okay, maybe let me just put the question in the chat directly and see if we can. Okay, somebody says it's kind of blurry. Somebody can see it, but it's blurry. Okay, let me just copy the question and just, because I wanted the post to be something that you can just, so I'm putting the question in the chat box. Or let me see if I could highlight it to the end. Okay, so just pick the number that is relevant and put it in the chat. So I'll make that chat something that is visible on the screen. Okay, so I'm taking the other one out now. So now this should work. So can you see it now? Just reply with a number. Sorry, some of the text were incomplete while I was typing, but at least you should be able to get what we're trying to say. So the most nearest answer or response, put it on the chat with your number. Let's see the number. So we're waiting. Can we all see it? I'm expecting the number you're going to pick. So reply with the number so that we will know the right answer. Just reply with the number 1, 2, 3, or 4. So So the options, the question is, what role does bioinformatics play to molecular diagnostics? It helps interpret, number one, it helps interpret the DNA sequence. If that's your own option, pick one. If it is two, it makes DNA sequence useful. Two. If it is three, it brings meaning to DNA sequence. You write three. Or four, matches DNA to their disease significance. Sorry, the space was limiting. That was why you see it like DX. DX means diseases. So it matches DNA to their disease significance. So whatever you think is the best answer, I could see one, I could see four, okay, four, beautiful, four. So there's about 2 4s, 1 1. Okay.
Have you joined the studio?
Okay.
Yeah, I can see you.
Yes.
I can see you.
You can see me. Okay, thank you.
Okay, so that's beautiful. So in all, for summary of that session, of that poll, the right answer, or let me say the most suitable answer is it matches the DNA to the disease's significance. Thank you so much for that. So, and I can see majority got that answers right. So now we are going into the session we've all been waiting for. And our facilitator is all set. And there's no other person than MLS Paul Orieji, FIISSN. So please, can you adjust your camera a bit, sir, so that you can stay in focus? Okay, thank you so much, sir. So now, to reach the citation is no other person than the Secretary of the Committee, MLS Yusuf Ahmed Rahima. Over to you, MLS Rahima. Okay, keep going ahead. MLS Rahima. We cannot hear you. Maybe your mic is muted, please. MLS Rahima, we could not hear you yet. So please speak to the mic now. Okay. All right, so if we are still having technical difficulty, I think somebody is going to be standing in, and there's no other person than the chairman. You know, when the secretary is having technical glitches, the chairman stands in. So, and the chairman of the committee, in person of MLS, Bello Muritala, will be taking the citation on our behalf. We hope is audible.
Good afternoon, everyone. It's a pleasure and honor reading the citation of our guest speaker for the second section, scientist Paul Orieji, FIISSN. Scientist Paul Orieji is an accomplished medical laboratory scientist, administrator, entrepreneur, and transformer. Permit me if I'm not calling those names well, Nigeria. His educational journey began at Agwena Primary School, Okwe Ogwo, after which he attended Ishagu High School and also at the Technical College. Driven by an unwavering passion for knowledge and excellence, he proceeded to Ebonyi State University, Abakaliki, where he earned a Bachelor of Medical Laboratory Science, BMLS. Even as a student, his exceptional leadership qualities distinguished him from his peers. He was elected president of the Okwe Student Union Association, served as the pioneer Igwe of the Faculty of Earth Sciences and Technology, and was Secretary General of the Federated Okwe Undergraduate Forum, leaving behind a legacy of visionary leadership and selfless service. Today, scientist Paul Orieji serves as the head of the histopathology/cytopathology unit at the Federal Medical Center, Abuja, where he has made significant contributions to diagnostic medicine, quality laboratory practice, and professional development. His dedication to excellence has earned him the respect of colleagues and professionals within and beyond the medical laboratory community. His outstanding leadership credentials include serving as the immediate past chairman of the Association of Medical Laboratory Scientists of Nigeria, Amlsn FMC Abuja chapter, and the immediate past chairman of the National Union of Allied Health Professionals, NOAP FMC Abuja chapter. He currently serves as the president of the Federal Medical Center Abuja Staff Multipurpose Cooperative, where he continues to champion accountability, innovation, and the welfare of members. Beyond public service, he's an accomplished entrepreneur and business leader as the Chief Executive Officer of Zenith Diagnostics Limited and Tabicare Limited, organizations committed Paul Orieji has participated in numerous professional trainings and has received several certifications and prestigious awards in recognition of his excellence, competence, and outstanding contributions to healthcare leadership and community development. He is a proud fellow of Institute of Industrial Security and Safety of Nigeria, FIISSN, a testament to his commitment to professionalism, safety, and national development. A visionary leader, mentor, and advocate for excellence, Scientist Paul Orieji continues to inspire young professionals through his unwavering commitment to service, ethical leadership, innovation, and nation building. His journey stands as a compelling testament that dedication, integrity, and a passion for impacting lives remain the true hallmarks of enduring success. Permit me to introduce once again and allow Scientist Paul Orieji to take the center stage for the presentation. Thank you.
Thank you so much for that, MLS Bello. That's an amazing one. And over to you, MLS Paul Orieji. Thank you so much.
Thank you very much. Thank you. I don't know whether I can be heard. Hello?
Yes, you're louder.
Okay. Thank you very much for this opportunity for we to interact and reason together as a medical laboratory scientist. Over time, we so much concentrate on definitely doing our work diligently, which is very okay, but we neglect the medico-legal aspect of what we do, probably because of lack of information or ignorance from the part of people that seek medical care from we that are health practitioners. But due to awareness and how people are beginning to know and seek for their right, it is pertinent for we to begin to realize that the work we do have some legal challenges. When we begin to have this awareness, we begin to avoid anything that has to do with compromise in discharging our duties. I've shared my slide. I don't know whether you can find a way to display it while I go on, but we will reason together. We are going to x-ray some certain things and divulge some information that are paramount in ensuring that we do not get into trouble as we discharge our day-to-day duties and activities. Cracking the code of silence, medico-legal reality.
Sir, if I may cut in, sorry.
Okay.
It's not sharing.
Are you sharing slide? Do you plan to share slide? Because I overheard you saying before, maybe why the share comes up or something.
I shared it to him. I don't know whether there is a way we display, because this place.
Okay. Yes, we can.
I shared it to him in WhatsApp.
Okay. So that will be resolved shortly.
Okay.
We'll try to understand. Thank you so much, sir.
I can go ahead.
Yes, you can go. You can keep moving.
Okay. So cracking the code of silence, medico-legal reality checks for the modern laboratory. The executive abstract and key terminologies which we need to understand. Clinical impact. Approximately 70% of clinical decisions rely directly on diagnostic laboratory test results. Positioning laboratory professionals as vital frontline decision makers. Over time, people like to downplay the role we play, especially our sister professionals, trying to ensure that people don't know we are behind the scenes. So, but the world is becoming aware of the role we play as medical laboratory scientists. The legal exposure, expanded responsibilities, creating heightened legal accountability. So negligence, poor documentation, confidentiality breaches, and critical value communication failures expose personnel and hospital to litigations. So when you see negligence, poor documentation, we keep on saying what is not done, what is not documented is not done. So when you begin to realize the effect of not documenting what you do properly, the legal implications and burdens you can bear when you did not document accurately, and confidentiality breaches. These are the things we are going to look critically today for you to begin to appreciate what you do and do everything to run away from legal challenges, in as much as we are doing everything to ensure that we get the accreditation, ISO 15189, splitter, and then all other regulatory agencies or accreditation agencies. We should also be aware the legal liabilities that we will always face if we compromise and if we engage in confidentiality breaches. And the code of silence, very important aspect of the topic that was given to me. Healthcare professionals frequently hesitate to report errors, unsafe practices, or misconduct. Overcoming this silence is mandatory to ensure patient safety, legal compliance, and quality improvement. So, code of silence. Healthcare professionals frequently hesitate to divulge errors. And I will tell you why as we go further. Why do health professionals hesitate to divulge errors, to expose non-compliance, to report issues that may be inimical to patients? They will shy away from it. As we go further, we know those issues and be able to ensure that we do not shy away from it. Core purpose. Corpus. This paper examines medico-legal responsibilities, identifies high-risk operational pitfalls, detailed ethical duties, and present actionable strategies to mitigate litigation's risk while reinforcing diagnostic excellence. Yeah, why this topic is very interesting and I so much love it is that in as much as we are trying to make sure that we take issues of litigation out of our way, we are also trying to also delve into ethical practices and compliance to ensure that we discharge our duties effectively. I think it's very wonderful and amazing at this critical time that the key terms and focus: medical laboratory science, medical legal practice, negligence and malpractice, patient safety, quality assurance, chain of custody, confidentiality and ethics, quality not sufficient. Terminological advancement and legal exposure. Every medical laboratory scientist needs a legal exposure. You don't need to be a lawyer to be legally exposed. You don't need to be a barrister for you to know your right, for you to know what you're supposed to do and what you're not supposed to do. And that is why our Council Act should be like your holy book. The regulatory agencies, other regulatory agencies are the accreditation agencies, their books, laboratory manual book, standard operating procedures. You should know them so that when we begin to see documents that can serve as an exhibit in the court of competent jurisdiction, you begin to appreciate your SOPs, your work aid, your attendance register, and other document papers you use in the laboratory. Because these things I mentioned can serve as an exhibit against whoever that engage in malpractice. Technological advancement and legal exposure. While technology drastically improves diagnostic speed and throughput, it simultaneously introduces sophisticated technical and regulatory vulnerabilities. Patients and families actively seek legal remedies when diagnostic errors. Okay. So patients and families, I think you can go back. That is where I'm at. Technological advancement and legal, technological, good. Thank you very much. Patients and families actively seek legal remedies when diagnostic errors lead to misdiagnosis, delayed treatment, or adverse patient outcomes. When we go further, you begin to appreciate that the sample you are holding in your hand, probably either from the ward or collected by we medical laboratory scientists, is a legal material. So you need to be very careful handling it. If you think that probably we begin to cover a lot of things, patients are beginning to be aware whether my brother or my sister died due to misdiagnosis, due to issues of malpractice from the laboratory, or non-compliance to standard rules and regulations. So we need to be informed, and that's why we are here today, to talk to ourselves, to inform. Scientific excellence alone is no longer sufficient for modern laboratory practice. No, scientific excellence. This guy is very intelligent. This guy is very smart. This guy is good. This guy knows what is. All these are no longer enough. Every specimen collected, every analytical assay executed, and every digital result transmitted carries significant legal weight and potential liability. So every specimen collected, every specimen, every sample, whether in histopathology, that tissue that comes to histopathology, that blood sample that comes to hematology, clinical chemistry, and that's two samples of fluid that comes to microbiology, has a legal liability, and it is very, very important. So understanding, you can move, understanding medico-legal practice and legal framework. Understanding medico-legal practice and legal framework. Definition of medico-legal practice: the formal intersection of medical laboratory science, ethics, and jurisprudence. It defines practitioner responsibilities, establishes standard of practice, and safeguards both patient rights and healthcare workers. Both patient rights and healthcare workers. And if there is any issue that warrant patient to go to court for any medical laboratory scientist or health practitioner, I want to let you know that attention moves to the patient that comes to court. Sometimes they give little or minimal attention to the health practitioner. They will focus to know whether this error is actually come from a health practitioner, because patient is saying it is due to the activities of this health practitioner that led to the death of my loved one. So the court, the judge, will keep on trying to know whether actually this error is coming from. So the attention is on you, and that's why you need to be very careful in what you do. Professional standards, guidelines, MLSN guidelines that we have. Sometimes somebody gets employed into a facility, employ you as a medical laboratory scientist. From there, you move to probably senior, principal, and chief. Now, let me even shock you. If you have any issue as a medical laboratory scientist, maybe let me just say senior medical laboratory scientist, and somebody decided to take you to court or to sue the hospital, and the matter gets to court, the court probably with their level of knowledge can ask for our scheme of service, and they bring the scheme of service as an exhibit. And who is this scientist that committed this error? And they look, is a senior medical laboratory scientist. They will first of all go to the scheme of service to know whether the duty he performed is the duty of senior medical laboratory scientist. When I begin to tell some facilities to make sure they discharge or they share responsibilities adhering strictly to scheme of service, it is very important. They will begin to question what you do. Is it in line with the responsibilities provided by the scheme of service? Sometimes the work of assistant director, the work of a director, you will leave it for a junior staff because you think, I have paid my dues. I don't need to, no, go and do it. When chiefs are done, they begin to ask, this responsibility that this person performed, is his responsibility? At the end of the day, if God find out that it is not a responsibility, therefore, he's bound to make a mistake. When you are performing a responsibility, a task that you are not trained, you have not been trained based on competence or whatever to do, and it results to patient death or harm, and you appear before the court of competent jurisdiction, the scheme of service will exonerate you and punish the person that's supposed to do it. It may not exonerate you totally, but the person that's supposed to do it, who was thinking that I have paid my dues and did not discharge the responsibility, we face it too. So we need to be very careful as we progress. Regulatory guideline, mandatory quality standard established by Health Accreditation Board and health ministries. Ethical principles, beneficence, non-malfeasance, integrity, and patient confidentiality. Please, am I being heard? Hello?
Yes, sir.
Okay.
Yes, you are.
Thank you. Thank you. Integrity.
Yes, you are.
Patient
You are being heard, sir.
Okay, no problem. Thank you.
Yes, you are being heard. You are being heard, sir.
I have heard you, sir. Patient integrity and patient confidentiality. Institutional policies, like I mentioned before, standard operating procedure, defining expert operational workflow within individual facilities, national statutory law, civil and criminal legal status, governing professionals, negligence, liability, and consumer rights, judicial precedents, case law, and court rulings. Why we, like you come to my facility today, there was the management wanted to force a pathologist on us. and we begin to resist it. And how do we resist it? We resisted it with our act, we resisted it with our scheme of service, and we resisted it with extant laws of the land, including court rulings from competent court of jurisdictions, and we said it cannot happen. We have some naked court rulings with us, which nobody should go against it except you appeal and they set it aside. As far as you have not appealed and they have not set it aside, even when you have appealed, this this ruling stands, and we will not allow any pathologists to come and hit the laboratory. And the sole reasons, if you want to go to court and vacate it, or go to court to set aside our act, or go to court to set aside our scheme of service that have empowered us to do what we do. Then deconstructing the code of silence in healthcare. You can move. I said that I want to tell you reasons why our colleagues decide sometimes to hide informations concerning those that are engaged in errors. Fear of punitive sanctions, litigation anxiety, workplace intimidation, lack of whistleblower protection, and professional embarrassment. Sometimes somebody don't want to be embarrassed, but you have engaged in something that you know definitely may cause a harm to patient. But because you don't want to be embarrassed, you want to allow it go. Not forgetting that allowing such to go may cause a harm to a patient. And when we say patient, everybody is a patient. You can be a patient. Your friend can be a patient. Your relative can be a patient. If you as a medical laboratory scientist can be a patient. So when we talk about this patient, so nobody should start excluding or exonerating him or herself. We are all patients in one way or the other. Because you may likely come to do routine tests. Not that you are sick, but you want to do a routine test. And somebody may give you a erroneous result. And that will put you in a panic mood. Or even lead to giving you a drug that you're not supposed to take. So when you begin to fear embarrassment, I don't want to be embarrassed by my colleague for revealing that I did this type of this thing. Said you allow such results to go. Lack of whistleblower protection. Somebody that blow the whistle, at the end of the day, you get punished. Probably by your organ, because the person that is engaging in this malpractice may be somebody, your senior. So you are afraid that you may not be protected. Workplace intimidation. People who intimidate, bully people. So you are afraid to be intimidated. Litigation anxiety. I don't want to, this litigation issue, so I begin to avoid it. Then you cause a harm. And the harm you cause can even lead to worse litigation issues. Fear of punitive sanctions. I don't want to be punished. I don't want to be punished. Then concealing mistakes. Concealing mistakes does not mitigate liability. It exponentially exacerbates it. When you conceal mistake, it does not mitigate liability. No, it does not. It causes more harm. It exacerbates it. And compounded patient's harm. Uncorrected errors propagate downstream clinical failure. Severe legal penalties. Cost view active concealment or failure to report as evidence of reckless misconduct, elevating negligence to punitive damage claims. Systemic vulnerability, unreported near misses guarantee future catastrophic errors. There's in fact well self-explanatory. When you don't report issues you're supposed to report and you allow it to go, it is catastrophic and will cause a very serious damage to such institution. So we need to be very careful to ensure that the code of silence is actually broken, and to ensure that we report things we're supposed to report. 4 operational phases of medico-legal vulnerability. 4 operational phases of medico-legal vulnerability. Now, when when you come to medical laboratory science, we talk mainly about three phases: the pre-analytical, the analytical, and post-analytical. And each phase has its own legal liabilities. When you come to medical legal issues, we begin also, we may not call it pre-analytical, post-analytical, but definitely they are interwoven. When you come to patient identification, you are talking about pre-analytical. When you come to specimen collection, you are talking about pre-analytical. Now you are talking about analytical errors, which has to do with analytical, and you come to post-analytical errors. Now, what are patient identification? How can patient identification be a liability? Legal liabilities. Wrong patient wristband or label, wrong label, that will lead to interchanging of samples. This is an honest mistake people do make that can land you in trouble. Switch blood tubes during draw. And that's why you must make sure that your table is clean and you draw one patient per person. You don't say I'm an expert. You label tubes. You bring patients. This one, you label two tubes for two patients together and begin to draw. After all, I can't make mistake. Before you could know it, there may be switch. So one patient per time, and you don't label your test tubes waiting for patient. These are mistakes that people make. You label your test tubes waiting for patient. Probably some hospitals, they write names of patients that are supposed to come for a draw. They give it to a phlebotomist. A phlebotomist will be calling name. Chukwudi Emmanuel. Before Chukwudi Emmanuel comes, because he's looking at the doctor's request form, the patients will do PCV, ESR, blah, blah, blah. He will now pick this thing, Chukwudi Emmanuel, PCV, or Chukwudi Emmanuel, whatever. He will label the container and be waiting for Chukwudi Emmanuel. It's very wrong. It's very wrong. Because you may call Chukwudi Emmanuel and Chukwudi Emmanuel is absent. If IUD will be called, and if IUD will call, you will know that you have already labeled Chukwudi Emmanuel. So what you do is that you label container when the patient is there with you, seated for draw. You ask him his name to confirm with the name you call, and he will mention it. Then you will begin to write the name of the patient in the specimen container. Or people that use barcode, you will know how to attach it. Sometimes they come with barcode already, sample that have barcode for you to draw. So all these things will make sure that you did not switch tubes during draw. Duplicate IS identification profile. Mislabeled transfusion samples. This is another place that needs a thorough check and a thorough put. Mislabeled transfusion samples that could lead to transfusion reaction. I see what some people do in a blood bank. And for those of you, probably you are operating in a blood banking unit. You need to listen and listen attentively. It is very important you pay attention. Now, some people do, they say they call it quick cross match. Quick cross match. A patient comes, even when it's not emergency or whatever, you will just do cross matching. You are not adding AHG. You are not cross matching with AHG. You will just quickly you cross match and release. Fantastic. Patient gets blood transfusion and go home and start having some reactions. And at the end of the day, if there is any hematologic reactions that led to death or severe issue, and they decided to go to court, the first thing that court will ask, your SOP, your standard operating procedure for cross-matching, and they will bring the standard operating procedure. At the end of the day, listen and listen very well for all scientists, so that I will tell you how this thing works. You will first of all ask for your attendance register. They have identified the patient, the scientist that did the work. Quite all right. The hospital have already identified the scientist that did the work. and the scientists that did the work will appear before the lordship, and they will be docked. Now they ask for attendance register. Attendance register is to ensure that you are on duty the day that thing happened. The attendance register will be provided, and the attendance register confirmed, yes, this guy was on duty the day the test was performed. They ask for license. This guy is a practicing license. This one will even come first. Court will scrutinize you. This guy, yes, is he a new? Is he a current one? Yes, it's a current license. He's a medical laboratory scientist. He has been licensed to do the job. He's not a quack. Well established. Is he on duty that day? Yes, he was on duty. He actually came to work that day, and he signed attendance register. Okay. Where is the SOP, the standard operating procedure concerning the job? And they will bring their standard operating procedure. The standard operating procedure will be before the judge. How was the test conducted? And you will begin to talk. Remember, the same sample, if actually it was a litigation that did not last for a very long time, and they are presenting some samples before the court of competent jurisdiction. How was the test conducted? And you begin to say, maybe your lawyer have guided you. Make sure you stated that you follow the SOP, the standard operating procedure. But you forgot that you registered the time that the patients brought sample from the ward for cross-matching and when the blood was released. Probably it is within 10 minutes. It is within 5 minutes that the arrival of the sample to the laboratory and when the blood was released for patient to be taken. When full cross-matching supposed to take, let's just say 30 minutes, 45 minutes to 1 hour. But the judge is looking at the prosecuting counsel has found a loophole that when they brought patient sample from the ward for cross-matching, and when the blood was released, it did not even take up to, because you called it emergency cross-match. The doctor is pressurizing you, release blood, and you quickly did whatever you did and release, and now there are issues. But you said you follow the due process and full distance. And we look at full cross-matching. Full cross-matching is giving us 45 minutes or 1 hour or thereabout. That case, you will go in for it. So no matter how doctor is pressurizing you, you must do the correct thing. Incorrect anticoagulant too. We just do it and think that this thing is not an issue, but it can land you in trouble. In vitro sample hemolysis, quality not sufficient, QNS, contaminated collection site, outcome, unreliable result, diagnostic delay. These are pre-analytical phase, but without the patient identification, specimen collection. In analytical errors, uncalibrated testing analyzers. The type of pressure that comes from management, forcing us to use machines that are not well calibrated, can cause you a lot of legal burden. When you are using a machine that is not well calibrated and documented that this machine is certified to perform so-so tests, but because once they supply a machine, we may not even care to calibrate it. As far as we are churning our result, you are producing one day wrong result that comes from that place may cause a harm that will lead to litigation. And definitely the court is asking whether this machine was well calibrated before running this test. You say yes. Can you bring all calibration documents for the court to see? You start running helter-skelter. So machine must be calibrated and documented before analysis will be done with such machine. Expired reagent and control used. You are using expired reagent to run a test. Some people call it revalidation. Let us revalidate it. It is still working. You are using expired control. Sometimes when they are moving control from abroad, it is there in a period before getting to your hand, it has already expired. And they will tell you control still works. 2 weeks, 3 weeks, 1 month after expiration, you can use it. You say, wow, you use it. Given the result passes, you are using it. Any issue that arises, and you come to court to tell court that the control that you used, though expired, but still working, you will not escape it. You will not escape it. So expired reagent must be discarded. When you say revalidation, you are putting yourself into danger. It may be working, but definitely the insignificant of it that you thought can cause significant harm to the patient that may lead to litigation. Ignore quality control, QC. Why are we even doing QC? This machine is tested and trusted. It is a durable machine. It's a Sysmex. Sysmex is wonderful. Big Makuta or whatever they call it is wonderful. These machines are durable. Whether you run QC or not, the test will always come out fine. That is, we are not speaking like a scientist and you may land in trouble. It's not human interpretation error. Human interpretation error. That is why we advocate. This human interpretation error happens mainly where scientists go in for it is private practice, where they interface with patients and explain results to them. But in a hospital, in a government facilities, we are not expected to interpret results. It is not our responsibility. I keep on telling this scientist, when we are trying to rub shoulders, we should know our boundary. Our boundary is the doctor say, I need this test to be run. Patients come, you collect sample, run the test. If it is where they are doing EMR or LIS, you send the result to doctors direct. Patients comes, you write your result, sign, and give the patient to go and see his doctor. Patient say, please, can you interpret it to me before I even see a doctor? He say, no. It is not my responsibility to interpret results. So if you venture into it and begin to interpret result, and along the way, there is an error that occurs due to wrong interpretation, you are going to go in for it if patients seek redress in court of competent jurisdiction. Avoid interpreting results in government-owned facilities. If you are managing a private, yes, it's understandable, but make sure that you know what you are doing before you begin to say, this one happened, this one, I begin to interpret results. So, human interpretation error can lead you into problems. Misdiagnosis, wrong treatment plan. Misdiagnosis, wrong treatment plan. You know, for private practice, they delve into treatment. Yeah, they recommend a drug you can take. Scientists do a lot of things. Ordinarily, they're not supposed to. You run tests, you say, go and meet a doctor, or you have a doctor you are partnering with, go and meet him to ensure that he gives you proper attention. I have finished my job here. But because we run private practice and we want to make money, we delve into areas that were not covered by our scheme of service and our expert laws. Our act does not empower you to treat. Our act empower you to do laboratory investigations to a patient. And we need to know that very well. Don't rub shoulder with anybody. When medical laboratory scientists begin to engage in treatment, you have gone out of the laws that empowers you to do your work. And when you appear before court of competent jurisdiction, you may face a truth that you won't like. Transcription, topographical mistakes, typographical mistakes. Typographical mistake we do neglect can cause a lot of harm in the laboratory. In some facilities, you see where woman was given prostate cancer just because a name was typed wrongly, and the patient collected it as her own and went to see a doctor, and to find out that the doctor saw prostate cancer, and the doctor was, what is happening here? This is not your result. And the lady has to come back to correct the correct one. Somebody that wants to make trouble will use that against such institution. So you need to be very careful while typing. So delay turnaround time. Delay turnaround time. If SOP, if your laboratory manual is saying, listen attentively. I don't know, somebody should mute his or her mic. There's a mic that is interfering with my own.
On this place is giving transmittance. Abi, the next one is what? Okay,
I think it's go to the next slide.
Transmittance in Igba absorbance, percentage percentage T. Absorbance.
Somebody is talking on the background. Someone is talking on the background, so it's affecting my. So delay turnaround time. If the standard operating procedure or laboratory quality manual stated categorically that EUCR turnaround time is 1 hour, and patients after blood draw comes 1 hour time and could not get it, come 2 hours time and could not get it, and at the end of the day the result was released after 3 hours, and it caused a harm to his patient, and the patient goes to court. Whosoever that ran that test will go in for it, because you have violated the code of conduct, you have violated the standard operating procedure, you have violated the laboratory quality manual of your own institution. It was not patient that made it. It is you that said their turnaround time is 1 hour. I don't have any business with that. But I came here 3 hours, the result could not come out. Whether if it's a problematic patient, whether that result, whether it caused harm or not, that person can sue that facility and say the delayed result led to harm to me or to my patient, and begin to seek and claim damages. So while writing your turnaround time, you should also be very careful and factor in some certain things. Do not do it to impress patient, and at the end of the day, you are not meeting up. It may land you in trouble. Patients are beginning to be aware. Failure to communicate critical values. Failure to communicate critical values. These are the areas I keep on advising medical laboratory scientists to work together with their chiefs and seniors, or even with the pathologists. If you have a pathologist in your facility, nobody is saying you should not flag critical values to them. You can flag critical values to them. You can flag critical values. It is very important to flag it, so that if you did not manage it very well, it could cause problem. Misrouted diagnosis report, outcome, sudden cardiac arrest, patient collapse. These are outcomes. Patient may have issues, and it can lead to a serious burden. Core legal principles: duty of care and negligence, duty of care and professional standard, legal expectations. Every medical laboratory practitioner owes a legal duty of care to patients to deliver services matching accepted clinical standard. Every medical laboratory scientist, no matter where you train, this should be with you always. Professional malpractice, defined as professional negligence committed by a licensed practitioner during clinical duties. I want to distinguish this. I want to separate this with quackery. You know, people don't know what is quackery. A licensed medical laboratory practitioner can never engage in quackery. You are not a quack. A quack is someone who is not licensed to perform a responsibility or duty. That is a quack. When you see a medical laboratory scientist engaging in malpractice, you don't say the person is practicing quackery. No. Once you are licensed and to discharge a function, and you are engaging in malpractice, we call it professional malpractice. That is why we can boldly say that pathologists who want to come and do routine tests are practicing quackery. You are not licensed to perform tests. And any facility they are doing that, scientists should begin to challenge them that they are practicing quackery. And this place is defined as professional negligence committed by a licensed practitioner during clinical duties. Such are deliberately reporting unvalidated results, bypassing QC protocols, or utilizing expired reagent knowingly. utilizing expired reagent knowingly. But I want to differ small here or to explain when somebody say utilizing expired reagent knowingly, which means you may unknowingly utilize expired reagent, but in court of competent jurisdiction is not admissible. Nobody accept that. But within our laboratory confine, it may be tolerated. But in a court of competent jurisdiction, as a medical laboratory scientist that can read leaflet and can read the body of reagent, you are saying you unknowingly use it. But let me tell you, so many laboratory unknowingly use reagents that are expired. They did not check. Reagent will be brought to you. You did not check, you begin to use it. You begin to use it. If any harm comes from that place, unfortunately, you may not even go to check the reagent you use, whether it's expired. And the judge calls, which method is this? You bring your protocols. You bring your protocols. Which reagent you bring? Can such reagent be submitted to court? And you people will rush and bring the reagent. It's a known reagent because court want to know whether this reagent is well known. Is it a standardized reagent? And they will look at it. Court is this reagent has expired. When was this test conducted? They state it. And they will check the body of the reagent and discover that the reagent you use has already expired. So what? That day the test was conducted, the court has found out that this reagent you use has expired. Definitely, every result that comes from this reagent will definitely cause harm. So you need to check your reagent very well before you begin to use it. Nobody will accept that you did not know. So duty, a legal obligation existed during professional to adhere to standard clinical care. Breach of duty, the professional failed to meet the accepted standard of care. Example, failure to report critical panic value, like what you say, injury or harm. The patient suffered demonstrable physical, financial, or psychological harm. Proximate causation, a direct causal link exists between the breach of duty and the resulting patient that injury occurred. Please go. 4 pillars of medical ethics. 4 pillars of medical ethics. The pillar of beneficence, the pillar of non-malfeasance, the pillar of autonomy, and the pillar of justice. I have to be moving fast because of time. Legal principles. Consent, confidentiality, and chain of custody. Very important. Informed consent. Acting in the best interest of the patient. So the first thing that will take you out of legal liabilities is to have at the back of your mind that definitely I should be acting in the best interest of patient. In the best interest of patient, not you. You can go out of your way to make sure that the best interest of the patient is adhered to. Concept emphasizing equality and distribution of resources among individuals. Justice, that is justice. What I just talked about is beneficence. In justice, you are talking about distribution of resources among individuals. Let there be no partiality. Sometimes management establishes VIP. Fantastic. They just want to make money, actually. In that VIP, they concentrate treatment and treat people because they pay double. There are so many ways court will look at that. There are so many ways court will look at that. Because if care is not taken, the court may be considering partiality in providing healthcare services, even why actually you want to make money. Yeah, it can cause little issues. There's partiality. The same facility, there is VIP, there is public. And in that public, anything goes. But in that VIP, because they pay more, you now provide essential service to them. If somebody is trying to report, because I'm public, I was abandoned. And I have a friend who I came together with because he went to VIP. In fact, he was given express treatment. In VIP, because I'm not VIP, that was why it led to harm. What they did to me now lead to harm, or death of a beloved one. So it will become a serious issue for such facility, and court will begin to investigate if there is any discrepancies for patients that enter such place. So justice requires there will be equal distribution of time, equal distribution of competence, equal distribution of knowledge, equal distribution of technical competencies to every patient that entered into a facility, to ensure that nobody is being treated badly because you don't have somebody or you don't know the medical director to talk to. 2, non-malfeasance, not causing harm to the patient. You are not causing any harm to the patient. Autonomy, respecting the patient's right to make their own decision. Now, I was telling somebody, do you know what is a patient? When the Latin word, they call it to become to say a patient. A patient is somebody who endures, who is having issues, endures harm, and endures issues or carry burdens, and the person comes to the hospital, and now the person enters phlebotomy or draw room, and you look for vein, you couldn't find vein, but you say, let me try. You try in one arm and you couldn't get it. You try the other arm, you couldn't get it. You say, please give me the other hand. He gives you the other hand, you couldn't get it. You try about 4 times. At a point, the patient say, please, I'm tired. What is it that you don't know your job or what? Why are you puncturing me up and down? And you flare up. Are you teaching me my job? This was somebody that had been enduring, just little statements you could not endure. So you are not worthy of that job you are doing. You should also put that patient, and it is wrong to put needle once and could not get it, put twice and could not get it, to go third time. It is wrong. Once you try, you did not get it, call a senior scientist to try. It is wrong. It is wrong. It is wrong, so that we will know what is wrong and avoid it. Once you try once and you don't have that confidence of going, call for a senior scientist. You do twice, please don't go third time as a person. Call a senior scientist to draw. Call a senior scientist to draw. If it is a child, please, you can ask a pediatrician to help out. It's just for you to avoid angering a patient. The legal defense and quality system, documentation. It is not documented. Legally, it was never done. The way we say in science, in our profession, whatever that is not documented, it is not done. I want to state that legally too, it is not documented, it was never done. Court rely almost exclusively on contemporaneous written electronic medical record to evaluate compliance with standard of care. So specimen accession log and rejection form. Let me tell you now essential legal record that may come to court when you have any issue. 1 is specimen accession logs and rejection form. 2, analyzer calibration and maintenance record. 3, daily IQC logs and out of control corrective actions. 3, critical value call logs, time reader and receiver. Incident and non-conformance report, CAPA. You will be hearing CAPA form, you are neglecting it. But when the time comes, you will know that CAPA form is a legal material. Staff competency and continuing professional education log. Staff competency training. Some will work from now till 3-4 years. They have never engaged training in any facility. See, you don't need to travel to Lagos. You don't need to come to Abuja. You don't need to go to Ghana to get training to be certified. No, you can organize internal training and document it. Attendance register, attendance list, and the training, document it. Because court will ask, has this guy been trained to do this work? And they say yes. It is only in court that licensing you to practice is not enough. Licensing you to practice, yes, is a general license to practice as a medical laboratory scientist. Performing tests. Have you been trained to perform these tests? Yes. Can you bring competency and continual professional education that make this guy competent to do this work. And you are looking, you don't have it. But you people have been engaging in internal training almost every week or every month, but are not documenting it. It is very wrong. Write an attendance, document the attendance, the training that was organized, document the slide. This one we are doing now is a wonderful training. Document it. The attendance, the people that is an attendance. So you will document it and keep it. It's a fantastic one to show that I'm aware that I should not do this, and I went ahead and did it. Now, internal quality control, daily verification of assay accuracy and precision, external quality assessment, interlaboratory proficiency testing, and comparisons. Are you people engaging in external quality control? Yes. Are you documenting it appropriately? Yes. How many times do you people do that? Well documented. Standard operating procedures, validated step-by-step technical instructions. Is there, many people always have a bench 8, no SOP. Just bench 8, they will attach it, no SOP. And some people, they have their SOP in a soft copy, not signed. Whatever that is not signed is not a document. In fact, if you have a safety protocols pasted on your wall and it's not signed, it's not a document. If you have work aid, you paste it. If it's not signed, it's not a document. Anything that is not signed is not a document. I'm not talking about the leaflet that comes with this thing, but you can see authority that wrote it. Anything you are generating from your lab as your internal policies, as your document, let somebody sign it and author it and own it. If it is not signed and you paste it on the wall, during the approval and they say bring it, and you are bringing something that is not authored, that is not signed, it is not a document. Court can never accept that as a document. And even accreditation and certification organization like Council for ISO 15489 cannot accept it as a document. So corrective and preventive action, CAPA, systemic root cause analysis of errors. Accreditation, international benchmark, proving clinical competence and quality, ISO 15489 2022. If you have it, you keep it. Now, other things I'm adding here is your attendance register. Your attendance register, very important. Your attendance register is also admissible before court of competent jurisdiction as a legal material. So it's better you note that. Ethical imperative and AI in digital diagnosis. Good. Ethical imperative and AI in digital. Can you move? Ethical imperative and AI in digital diagnosis. Beneficence actively promote patient well-being and safety. Very important. Normal patients do no harm. Prevent erroneous test report. Patients should actively promote patient well-being, and staff, medical laboratory scientists, or whoever that is a medical practitioner should actively promote patient well-being and safety. Normal patients do no harm. Prevent errors test report. Justice, provide equitable, non-discriminatory testing. Respect for autonomy, safeguard patients' rights, dignity, and privacy. Professional integrity, maintain absolute honesty in test reporting and quality documentation. Legal accountability, court holds human clinicians accountable for AI-assisted diagnostic decision. Automation is not a legal defense. Automation is not a legal defense. It's AI that is doing it. Court holds human clinician accountable for AI-assisted diagnostic decisions. So that thing you are using AI. Oh, AI is really. You are using AI to do it. Fantastic. But remember, AI will not be jailed. AI will not come to court. Court holds human being responsible for AI clinical decisions. Whatever a clinical decision AI does, the person who is manipulating or using the AI will be taught to answer questions. So do not think it is AI that everything is accurate. Sometimes AI engages in error. So checkmate everything as AI has come to stay, we should also be very careful on how we use it to discourage legal burdens and litigations you may not come out from. Mandatory human rights oversight. Pathologists and laboratory scientists must validate all AI-driven analytical interpretations. Now in histopathology, we are going into digital pathology where AI reads slide, AI does that and that. Now when AI reads slide, and it causes harm, is wrong diagnosis, as saying that somebody has breast cancer, and the person goes for mastectomy and they remove the breast, and at the end of the day, and discover that this person does not have any breast cancer, and it was AI diagnosed. Nobody who's AI accountable or responsible. The person who is using that AI goes in for it. So you need to be very careful. Deep dive. Insufficient specimen volume. That is where we are wrapping up this presentation. Insufficient specimen volume, QNS, quality not sufficient. Insufficient specimen volume, quantity not sufficient. Then, collecting an insufficient volume is not negligence by itself. However, how the laboratory handles, documents, communicates, or tests an inadequate specimen creates immense medico-legal liability. Medico-legal liability. One, delayed diagnosis. Insufficient sample. How do you handle insufficient sample? Now, sample come from ward for serum bilirubin or for PCV, parcel volume, for somebody who is having hemophilia, a baby that is having hemophilia, maybe is bleeding, and they take the sample to this thing to quickly do PCV, and it is insufficient for you to do full blood count. It's insufficient, and you could not even try to quickly do capillary PCV to at least get a result. Now, how do you report insufficient sample? What did you do? Every time these doctors, they always give us insufficient. Can't they collect it? You drop it and leave it there. And this is a test that's supposed to come out within 30 minutes or thereabout. And after 30 minutes, doctor sends somebody, go and collect result. And the person comes. You are telling the person, well, we did not run into it because the test is insufficient. You will sleep in quickly. If anything happened to that patient, you will sleep in quickly. This is the mistake so many laboratory does. You will just dump it waiting when the doctor will come. After 1 hour, doctor come. Please give us result, give us result, give us result. This patient was vomiting and stooling, or probably vomiting, and they rush to collect for EUCR to check for electrolyte and know how they can be able to. And they say it's magic, but it's insufficient. You drop it. You are busy. You did not communicate immediately back to the doctor that the sample you collected is insufficient to run EUCR. Please, can you resample, or can we only do electrolyte? So that the doctor will report back either resampling or say, go ahead and do only electrolyte. At least let's start from there. You just dump it, waiting for 1 hour, the doctor will come back. Delayed diagnosis due to insufficient sample that was not properly communicated and handled can put you in prison. This is the greatest mistake we do in medical lab. Immediately get back to reporting clinician. Do not drop any insufficient sample waiting for when they will come for the result. You will tell them after all that is insufficient. It's a negligence that could lead to patient death and could send you to prison. So immediately the sample comes, report appropriately to the requesting clinician that this sample is insufficient. And the doctor will definitely know what to do. Document it that I called the doctor and informed him that this sample is insufficient, check the time, write down the time of reception when you receive the sample. The time of accessioning is the different thing. Reception and accessioning is different thing. You access, you receive. You don't access what you have not received. The first thing you do, you receive a sample, you assess it. So when it's during the course of assessing, you discover that this sample is insufficient, and you document it. You call it the doctor, or you find a means of communication to communicate to the doctor that this sample for social patient is insufficient. You document the time of reception, the time of accessioning, the time, because somebody can receive and put you on a rack and did not access. So after 30 minutes, that's when you want to run all the badge. You begin to assess them. Very wrong. Once you receive a sample that comes from a phlebotomy or what, please go ahead and assess them. Right when the sample is phlebotomy. Because sometimes it's not coming from the ward, it's coming from the draw unit. And they are waiting for patients. They don't collect one and come, collect one and come. No, they collect everything together and now bring it to the lab. When sample gets to the lab, make sure that you report when the sample was received and assessed. I received it in 1005 and begin to assess in 1010. During the course of assessing, I discovered that this sample is insufficient to run EUCR and LBC. I contacted the doctor and told the doctor, the requesting clinician, that this sample is insufficient to run social tests. Should I go ahead to run only EUCR or full blood count? Because if we could complete this test, there must be a fresh draw from this patient. And doctor said, go ahead and do this. And I went ahead and did it. That was why you did not get the result for EUCR, or you did not get the result for FBC. You have exonerated yourself from any legal body. But if you keep quiet and kept and said anytime they come, or you did not state when you receive it and when you assess it, they will say that you received this since morning and did not get back to doctor. No, I did not receive it since morning. You gave it to a ward attendant, and ward attendant went to talk shop to go and eat before bringing it to the lab. And after eating indomie and the egg and whatever, after 30 minutes, he decided to come to the lab, and I received it. And you did not document the time you received it. The attendant will say, I knew he quickly came to the lab when it was given to me. He's lying. He received it when at social time. And you don't have any evidence to prove that you did not receive it that time, you will go to prison because you did not document it. But each sample you receive as a scientist and you document it, they will begin to question the lab attendant. This thing was given to you so time. Where did you go? They will begin to find out that you first of all went to talk shop to go and eat before bringing it to the lab. So it's very important. Pre-analytical prejudices, very, very key. Anything that has to do with pre-analytical analysis, we need to be very careful. And that is why in our facility, they say they train the scientific officers to be drawing blood. And we said, no, all of you get out of phlebotomy room. Nobody should be, professionals should be collecting samples, either our interns or technicians. These are professionals. They should know the sample code. They should know order of draw. Order of draw. Very important. You people do not know what is order of draw. You people do not know the code, what glucose the sample should go and all this and that, and how to even handle patient and the volume to collect. You see them collecting 1 ml for a sample, for a test, and at the end of the day, you are calling patient back for a sample and another. What if it is histopathology? In histopathology, after surgery, you are calling patient to come and do another surgery because you lost the sample. Or you are calling patient, hey, come on. Because let's is appendectomy that did appendicitis and they did the surgery and removed the appendix. Are you calling the patient come and remove which appendix again if you have lost the sample? So there are samples they will remove completely that don't have anything to remove. If the mastectomy, they remove the breast and you say, please come on, let's remove another breast. So reporting on unsuitable samples. Unsuitable samples, you worked on it and report. Documentation omission, we should avoid it. Medical legal case studies, the cause of procedural failure. Case 1, unlabeled specimen and workload. Listen to this case. Scenario, under high workload pressure, a laboratory scientist processes a blood tube with an unreadable label without verification. Outcome, result is assigned to the wrong patient, leading to a renal diagnosis and toxic drug administration. Legal ruling, breach of standard operating procedure, SOP, clear evidence of gross negligence. Moso and Okonkwo, 2023. This was a ruling. And everything I'm telling you is something that have come to court, and court have given ruling. There are so many litigations that is not reported. So don't think that what we are saying here, are you sure is happening? So result, the patients, something came and they printed, they did not print it, it's not well printed. And the guy did not verify it. He assumed that the name is, you are looking at it. You are saying this name is likely like Fashopo or what would be Fashopo, and you are looking at it. You say no. You call another person, come. You call 2 scientists, come. Let's put 2 hands together. Is this in Fashopo Aswaju, or is he Farotomi Aswaju? They look, no. So this thing is Farotomi Aswaju. You people will go ahead. No, the other one say, no, it is. My eye is very clear. You will run your test and give. Unfortunately, there is another Farotomi. And they treated the Farotomi instead of Fashopo. At the end of the day, this caused a lot of problem. So take away workload pressure. Workload pressure is never accepted by court as a legal defense for bypassing safety protocol. There's nothing like workload pressure. We had an issue here in our facility in blood bank, and I think what saved the person partially was workload pressure. But in a court of competent jurisdiction, court will not listen to that. That workload pressure caused you to make a mistake or cause you not to discharge the responsibility you're supposed to discharge. So when you engage in workload pressure, still make sure that you did not engage in something that will put you into trouble. So another scenario, a newborn blood sample for total bilirubin estimation is QNS. Quantity not satisfactory. The laboratory neither reject it officially in LIS nor request a repeat sample. They did not. They did not reject it through LIS. That's why LIS is fantastic. For LIS, you quickly just report it back that this thing is insufficient. But if you're not using a LIS, have a communication channel that you will quickly report it. So, officially in LIS, nor request a repeat sample. Outcome, severe hyperbilirubinemia progresses unmonitored to kinaseterus. That is affecting the brain, causing permanent neurological injury and cerebral palsy. Legal ruling, liability assigned to laboratory and facility for failure to communicate rejection, directly causing delayed treatment. Williams and Brown, 2023. So a sample came for direct bilirubin, and is an insufficient sample. It was not reported. It was not reported. And in the place, they told patient that you will come the next 2 days, or the next day, the next day. And the patients came next day, and that was when they were telling patients that sample is insufficient. Some lab will even go to the extent of discarding the sample. I am telling you, sample that says is insufficient, you discard it. And the doctor is saying, when the sample, they say, we discarded it because it's insufficient. Oh goodness. We have evidence to prove that this thing was insufficient. The doctor is hammering that sample, what is the volume that I'm supposed to collect? He says supposed to collect 3 mil of form, but that sample I collected is more than 3 mils. He said, no, it was 2 mil. I lied to you. We discarded it. You did not document it that it was 2 mil. There's evidence of documentation. You even went ahead to discard it. Now, even when we are saying that you're supposed to document it and you're supposed to call the doctor, you did not document it and even went ahead to discard it. And look at the severe outcome of it. Even when this thing did not progress to lead to kinesthesis or brain damage, the patient is claiming, the patient will that has said to that. Yes, the person that want to make it, if you go to loot, or you will see doctors, you will see lawyers hanging around, asking patients, were you well treated? Do you have any issue? Can we assist you? Don't even try and cry as patient and pass any of the doctor will call you, the doctor will call you. Is there any challenge? The doctor go and see what is happening there. They are well informed. Lagos, they are well. Gradually, it's trickling down to some places. Patients are not well informed. Lawyers hang around. There is no job. They just hang around looking for patients, asking patients, were you well treated? Is there any challenge? And they will see doctor because they will write to management and claiming damages, or we go to court of competent jurisdiction. So you need to be very careful. Silence and inaction on QNS, that is insufficient sample, carry catastrophic legal penalties. In fact, the judges frown at it. Court don't entertain that. You must communicate properly to requesting clinician. Standardize risk mitigation workflow for specimen. Inspect sample volume, anticoagulant or fixative ratio, and integrity before processing at pre-analytical logging. Inspect it, the volume, very important. If volume is inadequate, formally mark sample as rejected in LIS. Do not attempt on validating testing. Document exact reason, date, time, and identity of person rejecting the sample in electronic record or manual record. Document it. Avoid something that will put in trouble. Contact ordering physician. All this thing I explained to you is just what I'm reading now. Contact ordering physician directly via phone or LOS critical alert to state rejection rationale. Request priority recollection immediately to eliminate diagnostic delay and preserve patient safety. Request priority recollection. Request it. Don't not just stop by reporting. Request. That research should be done immediately. You have finished your work if they did not. Never process insufficient sample unless a validated, documented SOP specifically permits restricted testing with disclaimers. With disclaimers, if there is, people have an SOP on insufficient sample, how it is being handled. Every insufficient, there should be an SOP on how to handle insufficient sample. All this thing I'm telling you will be in that SOP. If you come to court and be telling court you did all these things, there will be, court will ask, is there any SOP that support your claim? And you say yes. Every facility should have an SOP for insufficient, on how to handle insufficient sample. Actionable legal risk reduction strategies for practitioners. Strict SOP adherence. Never bypass established laboratory standard operating procedure, regardless of workload pressure or clinical urgency. I have a workload pressure. Therefore, I don't want to screen this blood or do ELISA or whatever on this blood before transfusion. After all, we have done the surface screening and you transfuse a HIV that is on window period. You will go in for it. So insist that this blood must be screened molecularly, surface or whatever, to ensure that it's free to give to a patient. Not because there are so many work. I will not do cross-matching that I'm supposed to do properly. I begin to engage in shortcut and bypassing maybe AHU and whatever, and give the patients a blood that is not matching, and there will be a cross-matching reaction that may cause you trouble. Then rigorous identification, verification. Always verify 2 unique patient identifiers before specimen collection or processing. Those unique identifiers, if you come to histopathology, where we look at the sample bottle, we look at the form and check whether information on the sample and the form match, and check for clinician whether the number are fine, and everything we need to check before such sample will be accepted for processing. Meticulous audit documentation, record all corrective actions, calibrations, and phone communication with clinician contemptuously. Immediate critical value reporting. Promptly report critical panic values directly to authorized clinical staff and log call details. Lifelong professional competence. Engage in continuous professional education to maintain currency with modern diagnostic standard. Courageous whistleblowing. Promptly report unsafe laboratory practice, reagent compromise, or ethical breaches to quality. Institutional recommendation for healthcare facilities, quality management system, strengthen laboratory QMS, and achieve maintain ISO 154 accreditation to ensure structural compliance. Mandate regular medico-legal education. Regular medico-legal education. And that's why we're engaging with it. I'm so happy that people are beginning to be aware. In as much as you are going for laboratory quality management training and all these things, begin to go for legal training concerning what you practice, concerning this thing you do. Because even in teaching you medical legal aspect, delve into quality laboratory management. Everything you hear we say is what ISO is saying. We teach you more of quality management system than those that even train you on quality laboratory management system. Because it is the compromise of quality laboratory management system that will lead to legal burden. So we don't want you to engage in that compromise. That is why we delve into it more. So electronic error reporting system, implement automated LIS error tracking system to enable shameless root cause analysis, CAPA, non-punitive safety culture, transition from punitive blame culture to just culture framework, encouraging near-miss reporting. Establish protected anonymous reporting channel for safety and ethical violations. Conduct annual technical and medical legal risk evaluation for all bench scientists. Conclusion and the way forward. Cracking the code of silence, a moral and professional duty. Evolution of role. Modern laboratory professionals are accountable diagnostic clinicians whose decision directly impact patients' outcome and institutional liability. Breaking the silence. Overcoming the fear of reporting errors is essential to building robust safety system, protecting patients and minimizing legal exposure. Pillars of defense, meticulous documentation, adherence to SOPs, ISO 1589 QMS accreditation, and patient confidentiality constitute your strongest legal safeguards. The future standard, diagnostic excellence in modern medicine requires the perfect synthesis of scientific knowledge, legal awareness, and the moral courage to speak up. These are my selected references, and thank you for your academic attention. Thank you very much. Thank you. Over to you, the moderator. I think I'm done here. Thank you very much.
Can I be heard? Is it better now?
Yes, sir.
All right, thank you, everybody. That was an amazing one, and a very excellent session. However, before we conclude the session, there might be one or two questions that participants may love to ask. Please, let's go ahead and ask these questions. It's a question time. We have like about 5 minutes for us to ask questions before we round up the session. So if you have any question, please drop it in the chat. So if there's any question you may ask, so that we can tackle. Waiting for your questions. Okay. All right. So, no question. Does this mean that we have no question? Okay. There's a question here. Okay. There's a question. Go ahead.
Thank you for that. I want that.
Am I loud and clear? Can you hear me? No. Hello, can you hear me now? Did you hear the questions asked, sir? Did you hear the question asked, sir?
No, I did not.
Okay. MLS Bello, please ask that question again.
I said thank you for that wonderful presentation. I said in a case the MLS has a medical legal issue, maybe due to its practice, maybe he made a mistake that now led to a medical legal issue. I'm asking now that would that scientist go on its own to defend his case, or would have to contract a lawyer to do that for him? Did you hear me?
Hello, can you hear me?
Yes. Yes, we can hear you.
Somebody that is close to mic
again.
Somebody that is close to mic can rephrase what, can say what, because it's breaking, I can't hear her.
Okay, let me ask the question on his behalf, sir. So this is what the question he is asking. He said, okay, if a medical laboratory scientist have a legal case maybe there was a situation that a patient had to sue a medical laboratory scientist. What will be the legal action? Will the person go and defend himself, or he has to hire a qualified judge or lawyer for such sin? I mean to ask. So that's this question.
Okay, if I understand the question very well, it's saying if a medical laboratory scientist is having a medical legal issue, will the medical laboratory scientist just go and defend himself, or hire a lawyer to defend him or herself? Am I right? Hello? Hello?
You are right, sir.
Okay. In so many cases, nobody sues or takes just single out a medical laboratory scientist and take you to court. Most cases, they sue the institution. That's why they will write. It's the institution now will fish out the person who perpetrated that act. The institution will bring a lawyer to defend the institution. You that perpetrated act can also get your own lawyer. Is that you are now going to defend the institution and yourself, assuming that a blood sample, a blood transfusion was given to a patient, and there was a reaction due to improper or wrong cross-matching. The person sued the institution. You are not suing a scientist that you don't know what happened. You don't know whether you know that it's from the lab, or you don't know the person. Do you know who did cross-matching? Are you working with them? Are you in the roster? You don't know. What you know, you will sue the institution. And the problem is coming from the lab. The institution set up a committee to investigate what happened immediately, because they are going to court. They will get their own defense. And the institution will find a way, if it's a good, to defend their own. If they are not defending their own due to a known error, they will tell you, you will appear to court, you appear in court to defend what you did. You may hire a lawyer to defend you. Why institution is also have a lawyer to defend the institution of any error? Or the institution lawyer is defending both parties. So nobody sues Paul Orieji. No, the person does not know Paul Orieji. All the person knows that I went to FMC Jabi and so thing happened to my patient and I have an issue. I am suing this hospital for this negligence. You sue the hospital and MD that will go, and whoever the top management. What the hospital does, investigation, and find out that it is so person that committed this error. You will come and you will appear to defend what you did, either negatively or positive. So anyhow it goes. The person, at the end of the day, the person is now finding you, the court is now finding you culpable for causing such harm. So you go in for it, God forbid. So the institution is look at the person that caused this error, and we have been able to investigate that. So look at the procedure you follow. Now court is docking you and asking you, how did you do this work? What happened? Did you do this? Did you do that? Did you follow the protocol? Yes. Which protocol you follow? Look at it. Bring your SOP, bring this. Court is trying to know, was there any place infraction was engaged? Was there any malpractice was engaged? Was there any place something went wrong? If at the end of the day you followed whatever you did and the person still went, no, everybody is already, we call it, it's maybe error coming from, it's not from the lab. These people should check what they did. This guy followed every standard operating, every procedures to arrive to this. So most cases come from institution. Nobody knows the individual that happened, that caused the harm. They will just sue the institution, and the institution and take it up and bring the individual to come and defend what he did. Thank you.
For that answer. And let me just ask another question, please. There is this a common scenario that religious houses will ask the intending couples to do premarital screening, and they will expect that their results should be sealed and sent back to the religious leaders who ask them to come for the test. So considering the case of a confidentiality, what are the necessary documents that the laboratory that will run the test should have in case one of the couples or even both of them will later sue them that they divulged their results to the leader that sent them. So at the ab initio, what should be like a legal document that the lab should have to hold on to for obliging such a religious body's request? I don't know if you understand my question.
I do fantastically. It's a very interesting question. Very, very interesting question. The first legal document is what we call consent form. Consent form from such religious organization. Signed, and the consent form from the laboratory too. That consent form is either you have two or you make it two in one because you are partnering with such organization. Some of those hospitals, some of those religious organizations have a particular hospital they normally send based on trust. They normally send. So before accepting that, have a consent form. That consent form, one, in the religious organization, these people feel and agreed that our result should go directly to such religious organization after conducting it. And they will bring the consent form. When they have brought the consent form, signed by both the organization and all this, you bring your own consent form. Your own consent form is reading, I want to conduct so screening, marital screening or whatever for so both people. And they will sign such consent form in the lab that our result should go to social religious organization, not us. We permit it that it should be transmitted to them. So they will sign, the laboratory head will sign. It is a consent form in the laboratory and consent form coming from such organization. So you have both consent form that will exonerate you from breaching the cause of breaching confidentiality in terms of releasing result. And both of them sign. If you leave only the consent form from that of the religious organization to work in your own facility, they may deny that they did not sign such or they sign under duress. So when they come with such consent form, do not file it as the final document. Let them have a laboratory consent form where they will sign there, and their signature must match with the one they are bringing from the religious organization. Their phone numbers must match, their names must match. You staple the two consent forms and file, then you can boldly release result. The patient has given you consent. It's like signing a consent form that you want to be discharged. You sign a consent form against medical advice and you will be discharged. You are not holding doctors liable if anything happens when you go home. So get a consent form from the religious organization. Have your own consent form. You can fly, you can release your result because it's an adult consent. If it's a child, though it's now marriage, we are diving into if it's a child, the father or guardian will sign such consent form for searching to be. Confidentiality is very important. Thank you very much.
All right, thank you so much, sir. And that we are very grateful for that. There's a question that is being asked on the chat, and it has been pinned so that you can see. I don't know if you can see it on your screen
Yeah,
as well.
I can see it. What of religious organization that does not accept blood transfusion and the patient died?
Yes, in the process.
Okay, that is still a consent that somebody need blood transfusion and you are saying our religion don't accept it, we don't accept it. You bring a consent form. Sign against medical advice that you don't want your patient to take blood. Are we together?
There's another.
Yes, sir.
Sign a consent form that you don't want your patient to take blood transfusion. You are going against medical advice. You will sign it, kuro. There are people that do sign. I don't want any CS. My patient must give birth through normal route. So they will give you a consent form. You will sign it. You will sign it. When you sign it that you are against medical advice or blood transfusion, anything that happens is an adult decision. Remember, you will sign that consent. Presence of patient that's supposed to receive it. If that person is your wife, you are telling your wife, your husband said you don't want to take, you won't take blood. That is against it. Are you agree with your husband? You say yes. Sign consent. The husband sign consent. If the wife is not critical, she will also sign. It's not critical. If somebody is in ICU, that's when you will document that is in ICU. You can't be able to sign. Let the person that want to go also sign that they are against medical advice. You are exonerated of anything.
All right, sir. Thank you so much, sir.
Okay. Sorry.
You can go ahead, sir. The second question. That's another question.
What do we do in a case of insufficient sample from the ward and the doctor said it is what he or she can get from such patient? Can we go ahead and process it without SOP that support such and still write insufficient sample in our document? This is a case where patient is having an insufficient sample and doctor said this is all I can get. That is why I said there must be an SOP that explain how to handle insufficient sample. Now, a doctor, you call a doctor is saying, this is what I can get. You document it. Doctor say, this is what I can get. Doctor, should I go ahead and run this sample because it's insufficient? Go ahead and run it, please. I just want to look at what I can get out of it. We can't be able to get any blood again, is this. The doctor, both of you are trying to help a patient. There is no way. Doctor have said it can't draw. There is no way. But you have documented that doctor said I should go ahead and run this. Completely told the doctor the consequences of running, and he said, please let us look at it and know what we can do. You document it appropriately and go ahead and run it. And still make a comment, disclaimer, a comment that so mail of sample was received, which is insufficient for them. But doctor said I should go ahead and run it because it cannot get any sample outside this. And so result was said, you get a disclaimer, you release it. Doctor knows how he can handle it. But if there is any legal issue, you have your document and everything. And doctor, everything has to do with doctor. You say you can't get yes, and he will prove it. So you just find a way to exonerate yourself, but have SOP that deals with insufficient sample. Thank you.
All right, sir. Thank you so much, sir. And
thank you very much.
That was an amazing and fantastic response. So far, so good. So now we've learned, and I believe we all know now, the actions to take whenever we have insufficient sample. You are expected to still go ahead, but make your own documentation to guide against that. So thank you so much, MLS Paul and Paul Oreg. It's an honor to connect with you, and we so much appreciate your time. Okay, yes, I captured the FIISSN full meaning in your citation, but for the clarity or for those who are following, I think it would be okay for you to just state what that FIISSN simply means, because it was reflecting in what you taught us as.
Okay, FISSN means Fellow Institute of Industrial Security and Safety of Nigeria. I'm a fellow
Thank you so much.
of Institute
Thank you
of
so much,
Industrial
sir.
Security and Safety of Nigeria. Thank
So
you.
Industrial Security and Safety of Nigeria. You will see that he has really, really touched and taught this already. So we have come to the end of the change the camera. So we have already come to the end of the session, and we appreciate MLS Reggie for the session. And again, like the previous session, I believe all slides will be sent to our participants and our members. For anybody that also needs any special request or consultation with our facilitator, you can as well write to the email that was provided earlier, which will still be flashing through the screen right now. That's your screen, and ask contact@amlsn.oyshmb.org. So try to write to us. We will certainly reply as soon as we get the mail. Now, I believe this is a place to say thank you so much. Words cannot describe the hours, the attention, the professionalism that you showed in the teaching. And this is also extended to our earlier facilitators. And it's not yet complete if we don't also acknowledge our participants who registered and took their time to come for this session. And on this note, we come to the end of the webinar. We are very grateful to you all for your sacrifice. And you will all agree, and that is the value of knowledge, a well-refined knowledge. So sorry about the break in transmission. This is just all saying thank you everyone for your audience and staying till the end. I believe we have also gained not only 2 or 3, but a lot of information that could be utilized in our profession. And this also has increased our professional acumen. This will be the end, and this will be the place where we're drawing the curtain. We sincerely appreciate everyone. Do have a fulfilling afternoon or evening as we are looking forward to it. Goodbye. Thank you. Okay.